We were just waiting for you, Arunan, to say shoot.
Rahul. Oh, yes, yes, yes.
Kiran, I think we are getting some static noise from your side. Like it's constant, but I don't know what it is.
Is it fine now?
It's still there. It's some digital static noise kind of thing.
Okay.
Is it from all or only from Kiran?
It's from Kiran only.
Okay, I hope it is fine now.
Yeah. You may.
So maybe
Your voice is breaking, sir. By the way, we are live. We can start.
okay, do let me know if it is breaking still.
No, no, now it's
Okay. Good evening, everyone, and a very warm welcome to all the presenters and viewers for the Cube National Meet. So, we are celebrating 500 plus days of Cube Chatshala and Cube Home Labs. And if I say today is the 5-6 Inshallah. So today is the second day of National Meet, and yesterday we had the presentations on model systems like fruit fly, nematode, phyllanthus, and ants. The cubists are actually culturing fruit flies in homemade culture medium and trying to find out the species and conducting experiments in home labs. So nematode group is on the hunt of finding C. elegans of Indian native soil where they are isolating soil nematodes from soil sample on the potato slices. So we also had the presentation on Phyllanthus where Phyllanthus group is investigating the role of light and other external factors in external factors in sleep wake cycle. And not only this, but also finding out the different species of Phyllanthus plants. After that, we had presentation on ants, where cubists are interested in finding out how do ants communicate with each other and what is the genetic difference in the morphology of different categories of ants like queen ant, male and worker ants. And along with this model system, we had presentation on a citizen science project where we saw that we can be our own model system. And it is amazing to track the growth of the nail and investigate how nails grow and many more. So today is the second day of Cube National Meet. Cubists will share how are they sustaining research by finding alternatives to sophisticated experiments and methods in home labs. We are going to have interesting presentation today on earthworm, hydra, moina, and more. So stay tuned, and we will also be having a group photograph after the hydra presentation at 6:55 p.m. So let me add a personal note of welcome to the chairperson today, Rahul. Over to you.
Thank you so much, Kiran, and I hope I'm audible to everyone.
Yes, yes.
Okay. So welcome to the day 2 of Cube National Meet, and as Kiran already have told you about the day 1, it was a beautiful day 1 that commenced, and we look forward for a scientifically rich day 2 as well. And it's an auspicious occasion of Gandhi Jayanti as well, holiday, so would have a way to spend holiday than to discuss science and students of the country. So before we start with today's present students, I would like to tell you that these Cube National Meets is one annual event that we look forward to every year. So today we will be starting with earthworm as a model system. So earthworms, famously known as farmer's friend. Not only farmer's friend, but many cubists have also had a very intimate friendship with them, where they actually are trying to go into their lives starting from their birth, from their cocoon, to studying their developmental pattern, and also studying regeneration and asking a lot of other questions that cubists will be telling you in a short while. So earthworm is now a cubist friend also. And for today's presentation, without any delay, I would like to invite the earthworm group, which consists of the following cubists: Sachin Pradhan, Gaurangi Dhuri, Iram Bano, Saida Sayed, Ritik, Vaibhavi, Kiran Yadav, Nidhi Gavit, Saket Deshmukh, and Shreyashi. So over to you people, and let's start with our first model system for the day, and let's see what all good questions or science you are asking to this model system and what answer it's giving to you.
Can I start the presentation now?
Yes, Gaurang, yes.
So, hello everyone. A very good evening to all. So, myself Abhijit Singh. I am from Cube, Mumbai, Navi Mumbai. And as Rahul mentioned some time back, that today we'll be starting with earthworms. They are commonly known as farmers' friends, and it is very true that some of us also have kind of treat them as our friends, and we're also trying to understand some concepts like regeneration and other patterns with some kind of uh, ways and methods that we develop to how we can understand and work on that. So I'm sure that every one of us has seen earthworms. Like there is not a single person in this room who has not seen earthworm. I'm pretty sure that everyone has seen once in their life, and especially during the monsoon season. And it is almost an end to monsoon season in Mumbai, but still in monsoon season we see the earthworms, climbing in the walls or in the garden beneath the flower pots and places like that. This earthworm is very special. We have heard and also we have studied when we are learning in our school, as well as when we are learning about earthworms in school and all. We have heard that they are having in capability to regenerate cells. So it is very interesting to know that how this organisms, earthworms especially, are able to do that. So for that understanding only, we are developing earthworms as a model organism. So, I mentioned that in schools we study this, schools or colleges, when we study the classification of animals and the kingdom, then the phylum, and the taxonomy which we study. In that, we have seen earthworms as a reference to study the phylum Annelida. It is a very commonly in every textbook. I think there is mention when we are learning about phylum and Annelida, when they are showing an example, then earthworm is given first example. So what is earthworm? So actually, it is an organism which is found in earth, like the name suggests, earthworm. It is a worm and it is present in the soil. Mostly, it is found in soil, and its body is divided into segments. That is why I think it is placed in the phylum Annelida, because in that it is all the organisms which are having segmented body, they're placed in a phylum Annelida. And it is having a short lifespan of something like 40 to 60 days, around that. And also that is why these are the that is why this they are easy to culture also when we want to study on our question. So that is why we are developing earthworm as a model system. So when we talk about, like, when we have a question like we want to address, so for example in earthworms, as we are studying, we want to understand regeneration. So first we should have earthworms, so we need to culture them in an area. So we have seen that when we talk about culturing earthworms, people say that you need a vermicompost or thing like that, in that you can culture. But for conducting regular experiments or other stuff with earthworms, it is very tedious to keep to take earthworms from the soil and then clean them, and it is also dirty work as well. So just to avoid that, all of that sort of work, we cubists have developed the tissue culture. So we don't need any vermicompost and like that. We just use a tissue paper, and then inside a cup we just put tissue paper inside it, then wet the tissue paper, and just put the earthworm, and the earthworm lives inside this. And see, this is the slide in which we can see, this is the first and the second photo is from Ritvik. So this Ritik, sorry. This is a photo of Cube Lab, first and second photo, and the third photo is of Iram from her home lab. She's maintaining the earthworms in her home. So you can see how earthworms are eating and eating the tissue paper, and living there happily, because they are eating and defecating, and we can see all the defecated parts in the cup itself. So in the next video, in the coming slide, we'll see how to make the cup culture. So this is the video which shows us how to make cup culture. So first, we just take a cup, a normal plastic cup, and pierce some holes from the bottom. And this, what is the use of this holes? I'll be talking about that in some time. And this, we are now tearing the tissue papers, and we'll tear the tissue paper in short pieces, and in small pieces, sorry, and we'll place them inside the cup. So this video is actually made by Sachin. He's from Palghar, and he made this video showing us how we make cup cultures. So we just tear this newspapers, and we are putting them in the cup. Uh, yes. So now after we put the tissue paper inside the cup, we just sorry, we just wet the tissue paper with the help of water, and and then we just a little bit we try to push it inside the cup so that there is no uh, yeah. So next we are putting some earthworms in the cup, and then we will just cover the cup with the help of a cloth. And this cloth is just to avoid the chances of the earthworm getting outside of the cup. Sometimes they also climb the walls and they escape the cup. So this cup culture will be kept in a tray, and that tray will contain some newspapers or filter paper, and it will be wet with water. Then the holes which have been made, the water can through capillary action, the water from the tissue paper bed, filter paper bed, will go inside the cup with the help of the holes. So we don't need to moist the cup culture every day and open the, so that sort of thing can be avoided. So that is why we are putting the holes. So so this is how we make cup cultures in our home lab, in home lab as well as cube lab. Um, can we go to the next slide, please? So yeah, so now the question is that why earthworms only? Because there are several other organisms who are showing regeneration. So why? So as I said that it is easy to maintain and culture in cup cultures. Then it also has some similarities with humans. Like also they have cerebral ganglion. So ganglion, this term is actually used to show. So ganglion is actually a cluster of neurons that is present. So this acts as a simple brain in earthworm. Like we have a complex brain, but earthworm is a simple. Then earthworm is having peripheral nervous system and central nervous system like humans have. Then earthworms and human both have circulatory system. So with such similarities and easy to culture, we use earthworms. It is very easy to maintain, and also with the similarities with human, we can work on regeneration and other stuff as well. That is why we have chosen earthworms. Now, yes, the following part will be followed by Karangi.
Okay, so now we'll talk about our research questions. So as a cubist, we don't start any project as just take any model organism and start with the project. We need to have certain research questions, and then we continuously work on it to prove some hypothesis or something like that. So here in the earthworm studies, we have certain research questions. So everyone know that the earthworm perform this regeneration by there. So we have noticed that if we cut the earthworm into two parts or just cut the earthworm, then it eventually doesn't dies, but it starts to move. Both the parts move a little bit, and maybe after 2 to 3 days, it may be dying. So here we are studying. So this is the characteristics, that is the regeneration which is shown by the earthworms. So not only it regenerates when it is divided, when it is cut into two parts, but it also shows ventral nerve cord regeneration. It shows complete body regeneration, as I said just now. Then it shows immune response during regeneration. And one more thing, that is clitellum, which plays an important role in regeneration. So what is clitellum? Clitellum is the reproductive part of the earthworm. Clitellum is present in the 15th to 20th segment in the earthworm. As we all know that earthworm is a segmented body. Earthworm has a segmented body. So clitellum is present in the 15th to 20th segment of the earthworm. So these four are our research questions on which we are continuously working. So while doing the several activities, we discuss it in the Cube labs, as we know that Cube is collaborative understanding biology education. So we do everything collaboratively. We just don't perform any experiments and then write it and keep it with us, but we discuss with other cubists. We have certain discussions and then we come to certain conclusions, then we perform different experiments like that. Similarly, this is one of the Chatsala discussion. So here we discuss the story of earthworms and then how we can design an RER test. So RER test is a rapid escape response test to check the normal movement of the earthworm. So why to check the normal movement? So as I said earlier that our research question is to study regeneration in earthworms. So to check the regeneration, we should first know that the earthworm is doing its normal activities first, and then we'll work on the ventral nerve cord regeneration. So what we do in ventral nerve cord regeneration is just give us a little lesion on the ventral nerve cord, as we humans have the spinal cord. Similarly, the earthworms have the ventral nerve cord present on their ventral side. So if the human spinal cord is hurt, then we go under the paralysis. But that doesn't occurs with the earthworm. Earthworms also paralyzed for certain days, but it also regenerates its ventral nerve cord. So to study that, first we should check that the earthworm is doing its normal movements, and then we can perform a lesion to it, and then we can check whether the earthworm is actually paralyzing or not. And if it is paralyzing, then whether it is really regenerating its ventral nerve cord or not. So this was our Chatsala discussion. So here, as we discussed, we set up an experiment that is RER, as I said earlier. So what is RER? RER is rapid escape response acid. So we all know that if we put certain salt on earthworms, then it gets irritated and then it tries to run away from the salt. So why does this happen? So earthworm needs earthworm needs uh, earthworm needs moist surrounding and to live. So what if the salt is falling on it? So due to plasmolysis, due to osmosis, the moistness of the earthworm get transferred into the surrounding, and it can eventually die. So it tries to run away from the salt. And thus, by taking this point into mind, we discussed and we set up an experiment, that is RER assay, in which we are taking NaCl solution, that is salt solution, a normal salt solution, and we are putting our earthworm in it and trying to check that whether the earthworm is escaping it or not. If the earthworm is escaping, then we can say that the earthworm is normal. And we also take a control of distilled water to check whether the earthworm is escaping from distilled water or not. Earthworm doesn't escape from the distilled water because the distilled water doesn't contain salt solution, so that we can say if the earthworm is not escaping from the distilled water and it is escaping from the salt solution, then the earthworm ventral nerve cord is normal. So to support our question and to support our discussion, we cubists always search for different research papers in the net. So similarly, we found one research paper which says the restoration of sensory and motor function in earthworm escape reflex pathways following ventral nerve cord transplantation. So this tells us that the ventral nerve cord is restored by the earthworm. So this is one of the video of the RER in which we performed in cube. Like RER is rapid escape response, as I said earlier. So here I've taken 200 millimolar NaCl solution. As we were in lab, it was easily possible to take to measure the salt and to measure the salt and to take the NaCl solution easily. Uh, so here we have set the timer. We are taking care that we are not exceeding 2 to 3 limits, and so we standardized uh, up to 2 limits. We tried with 1 minute also, we tried with 3 minutes also, but we should also means we should also take care that the earthworm is not getting harm. If maybe it is possible that the ventral nerve cord is already destroyed, then is not well, then the earthworm can't be able to escape the NaCl solution. So we can't take risk that the earthworm will die inside it. So hence we set the limit up to 2 minutes, maximum 2 minutes. So here we'll see when the earthworm is escaping. So here we have set the timer. Here is the earthworm. If you can see, it is trying to escape. This is the NaCl solution trying to escape. And it will escape within a minute. So we were trying get to for 2 minutes. We also tried it in distilled water, but the earthworm was unable to escape from the distilled water. So here you can see the earthworm has escaped from the NaCl solution in just 1 minute 5 seconds. So now I would like Iram to tell how we perform RER in home lab. So as I said, it is a bit difficult to take the measurements and all, but now she will say that how we can do it in with minimum requirements in home also. So Iram, you please take over.
So, Gigi said you all about the RER that we learned in food lab, but because of this pandemic, we were unable to perform it at home. So, we all discussed in a chat shala and make a suggestion of it how we can do it RER in home lab. So, we all need a one measuring cylinder for that. So, in home lab, it is not available. Instead of measuring cylinder, we have used a cup which is available, and we have used a half tablespoon of salt, and which is approximately 5 grams. And we were not having a distilled water in our home, so we took a boiled water. I boiled the water and leave it until it cools down. And instead of test tube, I have used the plastic cups. And this is how I prepare the NaCl solution. I have used a half tablespoon of salt and half cup of water, and I dissolve the salt in the water. So I have. This is the video of Arya, how I am performing. This is the distant water. As we can see that from our distant water. And now this is a video of NaCl solution. As you all can see that earthworm is trying to escape from the NaCl solution. And now Saida Didi will continue the regeneration of earthworm after the amputation.
So with the help of RER, we could actually understand whether the cut on the ventral side affecting the ventral nerve cord, how is it, how the earthworm responds, recording the earthworm response with salt water. So what if we cut the earthworm in two pieces, like completely not just a simple cut of, but completely making it two pieces. So what we observe, can we have the next slide, please? What we observed is the part that had clitellum actually regenerated, whether it be the head or, so we took two badges. In one badge we had the clitellum with the tail part. So what we observed is the tail part is growing back the head, and another set we took, we had amputated the head portion. The head portion actually contained the clitellum, and in that set, um, the head regenerated the tail back. So we had a question whether clitellum plays any role in regeneration. And so for that, in Chatshala discussion, we actually found out, can we go to the previous slide, please? We actually developed a design that what we can do is cut up earthworm in three parts, as you can see at the right. Blue line clitellum, below blue line clitellum back amputation. We cut earthworm in three parts: one the head portion, one the tail portion, and one the clitellum itself, and see if clitellum can regenerate both parts. But of course, we haven't tried it yet, and we need to further discuss and develop more on the same. And can we go to the next slide, please? So Cube always has this culture of looking at the literature. So when we were actually looking for the role of clitellum, we can actually use any earthworm as of right now, because when we used native earthworm, even then uh, we were seeing that the earthworm with the part having clitellum actually regenerated. And Ritik and Vaibhavi, two of our collaborators from Mumbai, actually found this out in Acinetophyta, particular species of earthworm. Although this research paper, the literature actually says the amputated segment having the intact clitellum were able to heal the wound and form a regenerative blastoma. So even this paper, even the researchers actually recorded the same thing what we had observed. So we are still finding out, discussing about it as to how does clitellum play a role in regeneration. And with the same, we have the question whether what is the response of immune system to these regenerative properties. Sachin, can you take up that? So the thing was, during discussion what we found out, that okay, even the regeneration that is seen within 20 days, that head part, which is the part that has clitellum, is regenerating the other part back. Suppose the head has the clitellum, it is regenerating back the tail. The tail was not immediately dying, it survived for 2 days. So what actually happened? How did it survive, and why did the blastema formed? How things were happening, like, as in what was responsible for it? And in the discussion there was a point that okay, can there be immunity playing a role, because we come across immune system, studying immune system in our curriculum, and now with COVID, immune response and immunity is the common term that we come across. So does immune system plays a role in this, and if so, then how can we test it? So you can see at the side, we have talked about Gimsa stain as well. So we were thinking that we can take the fluid that comes out of earthworm while amputating, and check with this Gimsa stain, because it composed of both acidic and basic stain, to see whether what kind of cell it is, what is happening to the cell. But that is again still under development. We'll develop it through Chatshala discussion, through collaboration, and then work upon it. But when we again did a literature survey about the same, look at the literature about the same, we found that there was this paper called as injury-induced innate immune response during segment regeneration of earthworm. And so it is a different type of earthworm that we were working on, but they found that innate immune response is playing a role. So of course, we would like to continue study with taking this reference as as a key to understand it in a much better way and sort of through collaboration work on it. Yeah. So future plans, our future plan is to basically maintain cup culture. So as of right now, few of us have already started getting earthworms and started culturing it in cup tissue paper cup cultures, because and the long-term objective, again, you saw that we have several studies that we can do with earthworm. One is, okay, we can understand regeneration, we can try and understand, okay, how does ventral cord regeneration happen, does clitellum play any role, and earthworm as a whole gives an idea of different context to curriculums, covers several topics of our curriculum. When we talk about nerve cord regeneration, it is the study related to nervous system. How does nervous system get developed back? How does that regenerate? And even if you cut earthworm in two pieces, the whole body part is coming back. How is that happening? How till then, how is the earthworm surviving? That is the best way to sort of study regeneration and nervous system. And also the food, when we talk about the tissue cultures, they are feeding on cellulose actually. That's why we are using tissue paper, so we can understand that, okay, what exactly is happening, how cellulose breaking down, so we can understand biochemistry through it. So so there can be several different aspects of a curriculum can be studied throughout. Thank you. This is people from different our collaborators joining us from different parts of the country. Yeah, you can see Maharashtra, Thane, several places people are joining us regularly. Thank you.
Thank you, everyone. Now we are open for the questions.
Okay, so that was a very beautiful presentation, and because most of the cubists, like those who are today present here and have been a part of you from very long, must be knowing that earthworm always was a fascinating model system for cube. And I got reminded of my own undergraduate days when I also started with the earthworm and trying to figure out the morphological pattern of its regeneration. And it was very good to see all those experiments, starting from rapid escape response to nerve cord regeneration to the body axis regeneration. And we have a lot of questions from our viewers and also from the public. So to begin with, because you said that earthworm is very important and we understood why it is important from an experiments point of view, right? We can ask them a lot of questions and it can give us a lot of answers. But a question came that how do you think that outside the academic setup, how can we look at earthworm, and is it still beneficial? We know that it's farmer's friend, so it's beneficial for farmers, and we got to know that it's cubist's friend, so it's beneficial for cubists. But I would like to ask the team, anyone, what about outside the academic setup? Is still earthworm significant and important for citizens in general?
Yes, sir. It is absolutely very important outside the lab and outside the experiment basis, because earthworm eats the soil and then softens it and give the fishes, which is very soft soil, and it is also said as the farmer's friend. And it is this soil is very helpful for plants and other crops to grow. So it is absolutely very important other than lab and experiments.
So like for in general, people who are in Mumbai or Maharashtra or someone who's in Delhi, like we see earthworms during the monsoon. So another question was that why do we see all of a sudden lots of earthworm around us during the monsoon or when it rains? Where are they rest of the time?
Yeah. So as I said, as I said earlier, that the earthworms need the moist surroundings. So while the rainy season, the pits and all the grounds and all the the water gets gets entered into the soil and all, and in a large number of amount of water, the earthworm can't survive. So it just comes out from these things, and it finds a nice place to lay means to live, where it won't die.
So you mean to say that though they love moisture, but they cannot survive immersed in water?
Yes, it won't
What you're saying?
survive in immersed water. Even in distilled water, if we keep earthworm for a long time, it would try to escape. Hence, I said that we are keeping it just for 2 minutes and not more than that.
Yeah, so talking about soil and moisture. So another question that came from people is that, so when you were describing the culture setup, you were using tissue paper, right? And cubists have used tissue paper for culturing earthworms. But why are we not using the soil? Because this is one question that we begin with, and I'm sure that you must also have begun with the same question. They already live in soil; they have plants there. So why not use soil in a cup than using tissue paper? What is the advantage?
So, like when I was mentioning about the cup cultures, so like when we have seen in the presentation that we are doing RER.
Abhijit, just come a little closer to the mic.
Okay, now am I audible?
Yes, please.
Yeah, so like I was mentioning in the part where in the part which when I was explaining about the cup cultures, is that so the plus point which we have is that we saw that we are carrying out various experiments like the RER assay and some other that part. So when from the soil we take out the earthworms, we have to clean them. Soil is very dirty, so cleaning them is very tedious, and also soil and earthworm, it is hard to find out where is the earthworm in the environment. So like that
But
stuff
why
is
are we
there.
cleaning? Why are we cleaning them? Like
So when we clean the earthworm, if we are using soil in soil when we are cleaning it, then we are putting the earthworm in the test tube, for checking out in how many times it responds to a salt solution. And so if the we have not cleaned the earthworms, then the soil which is present on the body of earthworm will disturb the solution inside the test tube, and the test tube water, which is the solution which is there inside it, it will become dirty as well. So to avoid this all tedious work to so in cup cultures, in tissue cup cultures, we can easily find out that yeah, there is earthworm. So let's pick it up and clean it with just a little bit of water, and then we can use it for experiments. So it is a time saving. It can save us a lot of time, and that is why we are using tissue paper.
also because a lot of things that you said, just to condense it and like focus on it. Like one reason that the cubists have always thought of is because soil, though it has a lot of nutrients. So someone also mentioned nutrients. So would you like to tackle the nutrient question? Like there is nutrients in the soil, we all know. Is there also is the paper also nutritious to them?
Actually, the
Yeah,
actually
yeah.
the earthworms eat the cellulose from the paper. As means we always say that soil is nutritious for the earthworms, but what exactly the earthworm eats from the soil is the remnants of the leaves and fruits or whatever is falling in the in the soil. So earthworm eats that. So we also know that the paper is, or the tissue papers or the newspapers are made from the earth, means a tree's bark. So that contains cellulose. So the earthworms eat the cellulose present in the tissue paper. So hence that is also nutritious
Yeah.
for the
So the
earthworm.
nutrient question is tackled, right? We are providing them with nutrients, but it's also the point that why we are not using soil, especially for the regeneration experiment, because soil contains nutrients. It may also contain other pathogens, right? Bacteria or some infective agents. So if we are cutting the earthworm and putting it back in the soil, or you are like, is it one of your motives to avoid any foreign infection or any other thing apart from, so you want to keep the system sterile
Also,
is one thing.
so also it is like very easy to look at, as in especially the regenerated part. When you cut the earthworm in two pieces, like it is easy to sort of take a closer look at it, and so that is also one of the aspects like you highlighted.
Yeah,
And
so it basically provides the experimenter with everything an experimenter needs to conduct a clean experiment, right?
yeah,
The
yeah,
Yes, the visual clarity, and you're keeping it sterile, and also the food is ensured, right?
yes,
But as you said
And
that
avoid it
earthworms,
from
yeah,
any
yes, go ahead.
pathogen attack also, as you
Yes,
mentioned.
yes, right. But as you said that earthworms feed on cellulose, so there's one question by Arun that cellulose carbohydrate cannot be the only source of nutrition. What about proteins? Every system needs proteins. Have you thought about it? And it's obvious because if they are surviving and regenerating, for regeneration we needs protein, right? We are making up new things. So where is that coming from? Do you have any idea, or do you think that this is one thing which is very curious, which came out just now and we have to look at it?
I think it would be very nice if you look at it. We don't have much idea about it. We will surely try to find from where it is getting the proteins and other nutrients in it.
Yeah, right. Okay, so it's very interesting to see earthworm, giving us so many answers in terms of scientific knowledge. And there was one another very interesting question, that because we are looking at regeneration, regeneration in earthworm, and we have already known, we already saw the group talking about nail regeneration. So can you draw parallels between the two? Like, is the science of earthworm regeneration related to nail regeneration? This was one
So,
question. Yes.
so yes, we could draw the parallel, as in the sense uh, we know that stem cells would be involved in regeneration, and for the nail as well. Even if you cut the nail, the skin part of the nail, it will regenerate. The part will be restored, and similarly for the earthworm. But for other model systems that will be seeing today, even they will be talking about regeneration in them. But there is, I feel there is slight difference, but yes, there is similarity, as in, of course, stem cells would be involved. So yeah, that is what I have to say about similarities between the two. The common link would
Yes.
be the way the regeneration is happening.
Yes. And
And
it's always so. Yes, Gaurang.
and yeah, and I would also like to add that if we find what is exactly responsible for the regeneration of ventral nerve cord in the earthworms, then we can try to find that is that thing or whatever the tissues or whatever chemicals that can be added into humans so that paralysis can be avoided. So that also can be found in later studies as we go ahead in our work.
Yeah, so you're trying to connect the ventral nerve cord of earthworm with humans. It's a very big
Yeah,
leap that
spinal
you
cord.
are looking forward, but again, one small leap at a time leads to a big leap, right? So
Yes.
we here at Cube basically are trying to focus on a lot of things which are not the immediate objectives, but also in long run, how can we understand and contribute to science, right? And since you talked about ventral nerve cord, I would like to raise here a question by one of the audience, that how do you ensure that when you make a cut in an earthworm, the ventral nerve cord is affected and not any other major organs or tissues? Do we have to worry about it at all? Yes, Abhijit.
Yeah, so for ensuring that, so as the name suggests, ventral nerve cord is actually present on the ventral side. So, in this picture, we can see that the earthworm, the part of earthworm which is in contact with the soil, that part is known as the ventral side, and in that part the ventral nerve cord is present. So we'll just give a slight deep enough to ensure from our side that the cut is given to the ventral nerve cord, and the further in the front portion, the back portion are disconnected. Then with the help of RER assay, if the RER assay is actually a response assay to check if the coordination throughout the body part is working, so that the earthworm can move out from the initial solution. So if there is no coordination because of the lesion, so there won't be any coordination between the front part and the back part. So the earthworm won't be able to climb up. So with the help of RER assay, we can ensure that the cut is given to the ventral nerve. That is how we are tackling that question.
Okay, so
And
it's anatomically,
yeah.
yes, Gaurangi. Yes.
Yeah, and as he said that after giving the lesion, when we perform the RER assay, the earthworm, it won't be like that the earthworm won't try to escape the the salt solution. It would surely try to escape the NaCl solution, but its forward part and the backward part won't coordinate, and hence it would just it would just remain inside, but it would surely try. So from that we can prove that the lesion is given to the ventral nerve cord only and not the other major parts.
Yes, so you
The
have a
coordination
readout to
problem.
ensure that the ventral nerve cord is affected, and also through the anatomical positioning of it, you know that it's on the ventral side. So you are giving a cut which is deep enough to hamper the nerve cord, but not not hamper the earthworm so much that it gets separated into two parts.
Yes, sir. Yes, sir.
Yeah, and also because when we talked about the tissue thing, one thing that we both mutually raised up was that in our culture we don't want earthworms to catch infection from other pathogens present in the soil. But this regeneration has been taking place in soil, like for millions of years for earthworm. So, what do you think is preventing the infection in the soil where lots of pathogens and everything is present? Earthworm still regenerates.
Yeah, so that is the part, we will also also discussing on that. So, when we do the amputation and cutting the earthworm and set the regeneration, then it regenerates in the natural environment also it regenerates. So we were addressing the question of immunity. How does innate immunity plays a role in that? So like we are not having a conclusion like yeah this thing, but we are constantly trying to discuss in the chatshala every day and trying to get some stand on that. So we are continuously discussing about how innate immunity of the earthworm helps in that. It is definitely
Yeah, but if it's
sure.
yeah, but if it's experimentally proven, don't you think, because it will be the reason, immunity will be the concrete reason, because otherwise how can earthworm survive in a very
Exactly,
pathogen rich environment, right?
yes,
Yes,
yes.
yes, yes. And also you also mentioned one paper where you talked about transplantation. So someone just raised the point that we are not here transplanting any nerve cord back in the earthworms, right? So someone transplanted the nerve cord and saw that it takes away the effect. Now it can show the response. So would you
Yes.
like to just shed some light on transplantation versus regeneration for our audience to be clear?
Yeah,
Yeah,
transplantation
so
is yeah, transplantation is artificially transplanting the ventral nerve cord inside the earthworm, whereas we are studying on the regeneration, natural regeneration, which is shown by the earthworm it by itself, and we are not putting artificially any nerve cord in it. The nerve cord, the ventral nerve cord, is regenerated by the earthworm itself. So these are the two different things.
I guess the idea of showing the reference was that even after transplantation, 4 to 14 days after the transplantation, um, the function of actually like the earthworm was responding, as in it took up the transplanted part. Like it not like it did not die. It still survived, and it there was in this
So trying to put forth the point that earthworms are very robust systems, because in
yeah.
nature also. So because this property of regeneration which is present in earthworm, and so robust regeneration that earthworm shows, none of you touched upon the point that why they need it in their wild. Like, because if you talk about this, if someone just briefly talks about this, I think all our audience will be aligned that what is this very special thing about earthworm regeneration we are looking at and we are cling to. So in the wild, earthworms constantly need this property to survive, and
Yes, in the normal wild, the earthworm needs this regeneration property because it just stays in the soil. There are many small stones, pebbles, and there are also many animals which can host on the earthworm. So many times the earthworm gets means cut into two parts or more
yeah,
parts. So
birds being one of the very
yeah,
major predators of earthworms, and
yeah,
also because they are in soils and farmers are working to just buy agricultural tools, they can get cut into two
cut into two
parts, right?
parts. Yes,
Yeah. So this
yes.
also answers the question someone Ritik asked in the chat, that does an earthworm die after getting an injury when in the soil? So does it?
If
No,
if
right?
it is cut into, no, it don't get it doesn't dies in the soil. If it gets cut into 2 to 3 parts, it will
Yes,
still
yes.
regenerate it. Actually.
Yes. So it was a very beautiful presentation from all of you, the Earthworm group, and I would like to. I think we all should just give a round of applause to the Earthworm group. Very beautifully, you did the experiments, and you also told that what you wish to do in the future. So all the best for this model system and all the questions that it can answer in the future with our cubists, and also for Earthworm being so robust and the cubists being so consistent and a good friend of Earthworm. Okay. So thank you everyone. Thank you the Earthworm group. And before we move on to the next group, I would just like to mention this, that whenever a group comes, I will just introduce the group by their names, but it will be good if you, before you start speaking, just mention your name and the place where your cube home lab is situated, because it will also help us and others to get an idea that, you know, we are not just a small set of people sitting somewhere in one corner of the country, but we are very diverse, just like our model systems. Okay. So for Earthworm being regeneration being the center of the frame, we will now move on to another model system, which is Hydra. And I would like to call the Hydra group. And Hydra is also an interesting organism because it's known to biologists for its regenerative ability. You know, to an extent where people also have written that Hydra do not appear to die. They are biologically sometimes immortal, or they do not die of old age, because they can also reproduce through budding. And sometimes it has been written that they do not age at all. So a lot of interesting things about our next model system also is in the center frame, uh, along with regeneration. So with this, I would like to, and also for nervous system studies, Hydra is also used extensively. And so let's go and see how our cubists are using Hydra or being friends with Hydra to ask what kind of questions. Are they asking good questions enough? And is Hydra answering them? So for the next presentation, I would like to invite the Hydra group of Cube, which consists of Rachel, Manmasi, Batul, Hina, Saida, Abhijit Singh, Abhijit Vinod, Raj Patil, Shravani, and Shruti. So again, before you start speaking, just introduce yourself and the place you are situated at. Over to you, Hydra Group.
Hello everyone, good evening. My name is Manmasi Bek and my home lab is in Ranchi. I'm from, I have done my graduation from DSPMU Ranchi. So this plant-like organism is what we call famously as the Hydra. Although resembling a plant, this organism is a freshwater animal that attacks on small organisms like water fleas to feed upon. This academic organism is a popular example in books like NCERT biology, where we give examples of Hydra in regeneration studies. In this slide, we can see that there is a comparison between a diagram of Hydra versus a real Hydra that has been isolated in Ranchi by Rachel, one of the cubists. So here we can see the tentacles in the book versus the tentacles that we have found in the real Hydra that we have isolated. The Hydra belongs to the kingdom Animalia, which also which is, which includes common all the animals like tigers, lions, etc. It includes uh, it belongs to phylum Nidaria, which the children might be knowing that jellyfish belong to. So in the picture, you can see Rachel is collecting water sample to get Hydra from a pond in Ranchi. These are the pictures taken after the Hydra have different foods from the freshwater bodies. They commonly eat water fleas.
Hello everyone, my name is Abhijit Vinod. My home lab is situated in Kerala. Here we can see that cubist Arif explaining about Hydra to other cubists. What is notable about this picture is that we are still using the Arif's proposed method of culturing Hydra. You see, before this, we were culturing hydras using hydras were fed with artemia, which were not found in India and was costly because it had to be exported and was also a marine organism. And similar problems was faced by researchers all across the country. And and then it was suggested by Arif so that how about feeding hydras with which are actually found in the same environment as of Hydra and are found abundantly. And it was like a light bulb moment for us. Surely enough, we started doing it, and we found success in it. And many researchers also followed the same method of culturing hydras with using moinas, and they could find success in it. Here you can see a picture of hydra feeding on moina. And this is my home lab setup actually. Here I have taken a small aquarium tank, substrate composed of stones and some vegetation like hydrilla plant, as you can see. And you can see that hydras are very much found actually thriving in the environment. You can see the lining across the stone and on the plant.
Hello, I am Rachel Tirki from Ranchi and I'm pursuing Masters in Microbiology from DSPMU. So this picture is from my home lab where I used to culture hydra in dechlorinated water. So and also I used to feed with moinas.
Raman's lab in Ranchi is a collaborative initiative of three cubists, Rachel, Manmasi, and Manjual. So in this left slide, you can see a hydra and a crustacean. They are interacting. Probably the hydra is attacking. And in the right, you can see the hydra cup culture. It has the original water collected from the pond.
So this is the video from Shravani, Mumbai. So she's showing how we used to culture hydra in home lab. So she's taking dechlorinated water. So what actually dechlorinated water is that we keep tap water and we use them after the dechlorination happen as it evaporate. So here she took the moina culture. So she's taking moina with the help of dropper and she's transferring it to the dechlorinated water. And again she's taking the moina from the dropper. So she's using the phone flashlight to attract moina. So that they can gather at one place and it will be easy to transfer it. So after transferring it, she's taking the hydra culture. So here we can see she's taking the hydra culture. Again she is using the phone flashlight so that moina can gather at one place so that it will be easy to transfer the moina to feed the hydra. So this is how Shravani from Mumbai feed the hydra.
So, hi, I'm Saida from Cube Mumbai. So, when Shravani was actually culturing this hydra at her home lab, as you saw in the video that she's feeding moina to these hydras, so separately she was also maintaining moina culture in order to feed the hydras. And at one instant, it did happen that she lost all the moina culture, and this was the setup that was kept near her kitchen, near her window actually. And After few, so she had kept the bottle aside, and after few days, 1 or 2 days, there was some algae started growing in that bottle. Although she did not throw it away, after like 3 or 4 days, her mom actually saw that okay, some algae is growing, and there was some movement. There was some movement in the water. So what is it? And when she asked Shravani took a closer look at it, and she found that okay, moina is growing in it. So though it was a goof of that, okay, she did not carefully look at the moina cultures, did they survive or not. But from this goof up, we did have an Alexander Fleming moment, in the sense that we got an self-sustaining ecosystem for Hydra. You don't need to feed Hydra here. They will feed on algae and they'll grow, and you can just feed that moinas to the Hydra. So the amazing culture that is there in Cube helps us by just saying, accepting our mistake and announcing it, celebrating it. Also gives us opportunity to look at some of the instances where it can become an Alexander Fleming, and this is one of them.
So this is the picture of green hydra with a bud. This picture is taken by Batul from Kolkata. So she took this picture in her home lab. So here she kept the hydra without the feed for months. So even without the food, hydra survived for months, and it doesn't only survive, but here we can see that it was showing the budding also. So from the discussion, we came to know that the hydra was showing a symbiotic relationship with the algae chlorella. So here in this picture, we can see the green chlorella is all over the cup, and also the microscopic picture of the chlorella under the microscope. So here there are some questions regarding the symbiosis relationship with hydra, that how how these algal cells are controlled inside the host, and are there any genetic change in the endosymbiotic algae with compared to the free living algae.
Yeah, so we'll also with the collaborative discussion in Chatshala and with Meta. So there's one more thing that we noticed, as earlier we were the earthworm group did a presentation, they were trying to understand regeneration with earthworms. So here in Hydra also we observed that they are showing a kind of regeneration. But when earthworms were used as a model, when we amputate the earthworm and they cut the earthworm, then only the part which was having the clavicle that was regenerated. But when we cut the hydra from the middle, the tail part is regenerating its head and the head part is regenerating its tail. So this picture is the picture which is shown. This shows us that the head part is regenerating its tail part after 48 hours. And in the next picture, we can see this is the tail part, and it is regenerating its head part after 48 hours. So when we cut this hydra from between, like from middle region, then it regenerates into a two individual hydra. So how is this happening in hydra? So Man, can you try to explain the mechanism? Can you please try to explain the mechanism behind this regeneration in hydra?
So there have been a lot of discussion about the regeneration. So when we come across the term regeneration, there are certain cells that are associated with that, and that are the stem. So stem cells, in terms of stem cells in 1962, John B. Gurdon started the stem cells research, and he had done that on frogs and tadpoles. In 2006, that is around 40 years later, Shinya Yamanaka worked upon the stem cells of mice, the skin cells of mice, and he turned them into the stem cells. The mature cells turned into the stem cells. So you can see that worked upon the induced, uh, they worked upon the inducing of mature cells into the stem cells. But when we come across the Hydra model system, the Hydra model system already contains the stem cells, which can be studied further to unveil various concepts. Talking about the objective, we have three main objectives for now. Those are to culture Hydra, to study regeneration in Hydra, and also to study the symbiosis in Hydra. By, by using Hydra as our model system, we came across several concepts from our curriculum. We came across taxonomy. We learned where to place Hydra in the ecosystem. And we learned about the ecology, where we learned that what is the role of Hydra in a food chain and the ecosystem. We learned that Hydra and Moina cultures both can be both are can be cultured from fresh water bodies, and they can be cultured at home. We came across the behavioral biology, which is which makes it Hydra is a very simple organism, and to study behavioral biology, maintaining very large organisms are very tough. So Hydra becomes a very important, very simple organism for the behavioral biology. Also, as we have gone through our textbooks, that Hydra is an example of regeneration. So it makes a regeneration example as well. So as Rahul has already explained about the neurobiology and developmental biology, so these are the concepts that for now we are covering, and we are looking for more concepts that could be covered. These are the collaborators list. Starting from the Ranchi to Mumbai, there are several from across the India, Kolkata, Kanpur, Kerala, Delhi. So our collaborators are distributed from across the nation. So this was our presentation. Thank you.
Thank you, Man, and I must say that what a commendable work you have done to take Hydra out of an NCRT diagram and to do a full-fledged undergraduate research about it, and telling us so much that textbooks cannot tell us in detail. So that was an amazing presentation, and the audience was also very much glued to the presentation, and a lot of questions came, a lot of which you answered in your subsequent slides. Those slides were very well made as well. But to begin with, there's this very basic question that how do you catch Hydra for culturing? Where do you get it from?
Hydra can be, we can get Hydra from the freshwater bodies like ponds, lakes, and rivers. I'm not very sure because they have a very fast flowing water. So
yeah, so the point is that you have to, like, what are the certain characteristic points for the water body where I should go, or someone like Kaninika asked this question, so she should go to find Hydra. Where should we go? What kind of water body should we look for?
Water bodies with like some aquatic plant where like abundance of aquatic plants are there, so that should be a good place to start looking for hydras.
Okay, and there's also another question which asks that why are you using dechlorinated water? Is it like the chlorine is harmful? What is it about the dechlorinated water, which, because you mentioned dechlorinated deliberately, so that raised.
Yes. So when we're talking about dechlorinated water, in our home, in tap water, the water which is coming is with chlorine. So in natural environment where hydra is found, like lakes or ponds, the chlorine is not there. So what is chlorine actually doing is that it kills the bacteria and other stuff, so that we cannot get harmed by it. But
these organisms won't survive in the chlorinated water. So that is why we are dechlorinating. And also it is important that when we are feeding the hydra with the moina, we are trying to feed. So that is also important. Moina is also not able to survive in the chlorinated water. That is why we are using dechlorinated water. So we just keep the normal tap water for overnight with open or with a cloth, and the chlorine just gets evaporated by that time.
Yes, right. So chlorine is an irritant. Basically, we use it to make the water potable. But yes, and because yes, we were seeing that Rachel is actually trapping Hydra, right? We saw this photo. So it would be nice if she talks about her experience of trapping Hydra from the natural source, because the first-hand experience always educates the most. Yes,
Yes.
Rachel.
So what actually I did that I used a plankton net. So this is actually a net that is having very small pores, and at the end of it there was a container that was actually gathering the water after filtering it. So this is how we can get more small
So
crustaceans or organisms.
Rachel, it will also be very good if you tell us that. So which is this place, which is this water body, and what pore size? Because you said small pore. So for someone, something, and what is actually the size of Hydra that we are capturing? So something, if you'll put more context to it, we'll be able to reproduce it. Someone will be able to reproduce it, right? What is it called? Is it called something?
So this this is the picture of Jharkhand fisheries, from the Jharkhand fisheries. So I I got this plankton net from them. So the pore size was less than 1 mm. So so from that net I collected the water sample, and in that water sample we got the hydra. And talking about the pond, so this pond was actually the natural pond that was made to culture the fish. And I must say that in in so many places that we see that we use the pond for washing clothes and all. So if we are looking that kind of pond, so we should look that site where we are not actually using soap. So because there uh, there can be less organisms, or the soap can harm the water body organism.
Yes, right. So the same thing about chlorine. Along the same lines, we should look for Hydra in places which are actually naturally not contaminated, right?
Yes.
Okay. Okay, that's good. And also in one of the experiments when you were showing, so it intrigued people where you showed that you put a cell phone light on, or the flashlight on, and that is a very easy way to catch Hydra, because a lot of Hydra are concentrated in one narrow region where the light is coming from. So they are, yes. So in this picture you can see they are attracted towards the source of light, right? So the question which arises is that why are they and how are they attracted towards light? Do they have something like eyes? Can they see light? And if they can, how?
I think the picture showed Moina being attracted towards light. Moina was being fed to Hydra, so the flashlight was being used to collect Moina at one place.
Okay, someone also asked, do hydras are also attracted to light? Like, are hydras attracted to light, and can we, in the same manner, can we actually make them concentrated one region?
No, when we flash the light at Hydra, the Hydra does not get attracted to it.
But it's very intriguing, because we know that Hydra has been used is known for its attraction towards light. Hydra actually can sense light. So, what do you think must be the reason that you have not seen that kind of attraction from Hydra in your culture?
One thing being in Cube, I believe, is you don't get answers to things if you don't ask questions. If you actually, we did not look for whether they are attracted to light or not. That is why we don't have an answer to that as of right now. But as you were saying, so I think this, because of this question, we would also be interested to look at whether they are attracted to light. If so, how and what
Yeah, because
actually.
yes, because in the beginning also, and when a question came in the chat, that also I connected that. I asked that let's see what kind of answers our cubists are getting from Hydra, and again it depends on what kind of questions you're asking. So Hydra is a very good example where they are known for showing their attraction to light even without having any kind of eye-like structure or photoreceptor cells in general. So that will be a very interesting question to take away from this session. And thanks to the person who asked this. Okay, so just in the end, I would like someone to just summarize, because there are a lot of things with Hydra associated. So a lot of questions that came were about that there is Artemia involved, there is Moina involved, there is Hydra, and there is Chlorella. So Chlorella is living inside the Hydra and Moina is being fed to the Hydra. Earlier it was Artemia which was being fed to the Hydra. So all these organisms, and they do not come from the same kind of like habitat, same source. So there was one question in between which says that why do we need to wash the Moina before you feed it to Hydra? And the same also must have been true for Artemia earlier when we were using Artemia, right?
So, why we wash? Why we need to wash Artemia when we are feeding it to Hydra? Because Artemia is not a fresh water. It means it belongs to the marine water, and Hydra is a freshwater organism. So if we are not washing it, and if we are directly feeding it to the Hydra, so it may harm to the Hydra. But in case of Moina, when we are feeding it to the Moina and we are washing it, so why we are doing this? Because when we are culturing Moina, so we are actually feeding the Moina to with the milk, and there can be debris in that culture. And when we are feeding Moina to the Hydra, uh, debris can be transferred. So we don't want debris with Moina. So that's why we are just washing it and then we are feeding it to Hydra.
Yes, okay,
Technically, it
perfect.
is
So,
not
yes.
washing. Like we are not washing the Moina, and like giving it a rinse of dechlorinated water. We are not doing it. We're just transferring the amount of Moina that we need to feed to the Hydra. So to another container, so that it is easier for us to feed the Hydras.
Yes. So basically, I wanted someone to clear that it's the Artemia we need washing for, not the Moina, because the question came from Moina, and because there were so many organisms associated in the culture, so might be audience oftentimes gets confused, and so it's important to understand that Artemia needs to be washed, because as Rachel said, it's cultured in a very high saline environment. It's a marine organism, right? And for Moina, we can even if we are doing some sort of washing, it's for the debris and not for salinity.
Yeah, even if you put Moinas as it is, it will not harm the Hydra, because they belong to the same ecosystem, as in they are all fresh water and they are both cultured in dechlorinated water, so it's not gonna harm them. Whereas Artemia, it's like marine water, so the ecosystem is sort of different. So that was the idea of washing Artemia, and I guess there are too many organisms involved.
Yes.
So that I would like to summarize a little so that the doubts are being cleared. That the symbiotic association, the symbiotic relation that we were talking about was between. I'll change the slide as well for the same.
Yes, it will be.
Was
Yes, it will be good if you explain all the four organisms and where they are actually in
yeah,
the end. Yes.
yeah. So hydra in green hydra particularly, there is chlorella present. So there is symbiotic association between chlorella and green hydra. So that is one thing. And then moina is actually feed of hydra. So yes, they can survive without feeding them. They can survive, but in the natural environment, they feed on moina. Sorry, they feed on water fleas. It's not specific to moina, and moina is a water flea belonging to the same environment. So we feed them moina. Yes, and earlier, before we could know about moina, um, Arif's point actually, we used to culture it. We used to culture Hydra with Artemia because that's how the researchers used to culture and research its teeth. And it used to be costly as well. And maintenance was also difficult, like washing it.
I think one point of confusion also must have arisen from the type, like the legend that you have written, that Arif showing Hydra using flashlight. So people
Yeah,
must be thinking that
that would be confusing. Yeah.
yes, Hydra is being attracted, and that's why they ask the question. Where it's true, because Hydra is attracted to light, and it's very intriguing and interesting that we have not seen it, or maybe we saw it, but we never
Look back and ask whether is it
never
attraction.
paid attention to it.
Yeah, yeah. So yeah, that is the key. And yeah, so that because of RF actually, it would be the simplest question for anyone, that why are you using two organisms from two completely different ecosystem? Why don't we feed hydra something what would be very natural to them, what they'll be feeding on in ponds and lakes? So yeah, that question actually changed the way we used to culture hydras. I think I have sort of accommodated all the organisms, and we are sort of clarified.
Yes, thank you, Sada, and that was required, and it was a very beautiful presentation, and a lot of good questions asked, and a lot of good experiments done, and we look forward from our Hydra group to unravel more mysteries about Hydra and a lot of questions, and especially the one thing that we are taking back from today's session is that seeing whether, how, why, and if Hydra shows attraction towards light, and that will be very interesting. So I would like to thank the whole group, and it was again, I think they also deserve a round of applause. It was very well led. And with this, we have finished two of our model systems for today. And I think this is the time where we will take a short break and we'll organize ourselves for a group photograph, right? Kiran will.
Yes, Ashutosh. So yes, those presentations were very nice. So here we can take a group photograph. If it was an offline meet, so we would have been standing in the HPCC garden and taking photograph. But since it's virtual,
I think
so
you put it very calmly. If it would have been an offline mode, we would have run towards the most open place that we would find in HBCSE. Most of the time, it's the center place, and everyone wants to stand in the first queue.
yeah.
Well captured. But anyways, what are we doing now? So we'll be.
Yeah, we are going to open our webcams. Everyone to open their webcams. I will do mine as well. I will call out names as well. Aswati, Saida, Abhijit, Anshu, Arunan Sir, Arunima, Batul, Dhanraj, Drishtan, Ganesh, Gaurangi, Hazel, Hina, please on your webcams. Ishita, Isha, Janvi, Karinika, Manju, Neha, Chitralekha Pam, Rahul, please on your webcam. Raj, Rachel, Sachin, Sanjeev, Shivam, Shreyasha, Shrutika, Siddhi, Soumyadeep, everyone, please do. Sridevi ma'am, Sudhir, Vyom Gupta, Zara. So I will start taking the screenshots. Please join us. Yeah, we can accommodate more webcams. Yes. We are missing others. Chitralekha ma'am, Hina ma'am, Aswati, Drishtan. Thank you. Yeah, Anshu is off me. Thank you. Thank you so much, everyone.
Only four of us.
Thank you so much, everyone. So we got the shots.
So, so shall we proceed again towards next model system, Kiran?
Yes, perhaps. Yeah.
Okay, so with these two model systems that we've seen, earthworms and Hydra. Uh, so with earthworms and Hydra, we saw that what a model system based research can give us as an output. And yesterday also we saw a lot of model systems giving us answers. And so moving from earthworm and to Hydra, now we are going to another model system, which is Moina. And we have already heard about Moina as Arif decided one day, and the cubists discussed and agreed that Moina should be the food for Hydra. But I would like to put here that one organism's food can be another organism's model system. So Moina can be a food for Hydra, but Moina is a very beloved model system for a lot of cubists, including one of my close friend Kajal, and we used to call her at one extent mother of Moinas. And she was attached to those Moinas so much, and I'm sure that a lot of other cubists now are walking in her footsteps, and they do not see Moinas merely as food for any other model system, but they see Moinas capable of giving them answers for a lot of different questions that other model systems cannot answer, right? And I'm sure that the upcoming group, the Moina group, is going to uh, make Moina look like a celebrity in your eyes, because till now it's just a food for Hydra, but it's not. It's much more than that. So Moina is basically is a crustacean, and it's also known as a water flea. It's very closely related to Daphnia, and it's very interesting that uh, for a long time, Moina and Daphnia were confusing terms in Cube, and I'm sure that someone from the Moina group will unravel that confusion also for us. So let's go and see what Moinas are good at and what Moinas can answer, and how are they just not a food, but a very important and a valuable model system. So I'm putting it over to the Moina group. And for the Moina group, I would like to introduce the Moina group. Uh, it consists of Drishtant, Ishita, Raj, Saida, Manmasi, Rachel, Anshika, Zahra, Kaninika, and Abhijit. So again, when you start speaking, please introduce your name again and your place, your location. So over to you, Moina.
Good evening, everyone. This is Anshika Pegwar. A hearty welcome to all of you present here. Today, we are going to present in front of you all our model organism, Moina. So, without further ado, let's talk about what is Moina. Moina is a freshwater crustacean which is found in fresh waters like ponds, lakes, and rivers, and is also known as water fleas. So, before we go further, I would like to show all of you a video about how we collect Moina. In this video, we can see Rachel collecting the water sample from a pond and observing it for the any movement of water fleas. So next, Moina, it feeds on bacteria, yeast, algae, phytoplankton, and detritus. The average length of Moina is zero point 0.7 to 1.5 millimeters, which is approximately 700 to 1500 micrometers. Average lifespan of Moina is around 12 to 14 days, in which the average life cycle of it is about 3 to 4 days. So here on the right hand side, we can see the taxonomical classification of Moina. So Moina belongs to kingdom Animalia. It is from the phylum Arthropoda, same as that of fruit fly. The subphylum of Moina is Crustacea. Uh, the crab, as we can see in the picture, crab also belongs from the same subphylum. It shares the same class as Artemia. The class is Branchiopoda. Order of Moina is Cladocera, which is shared by its close relative Daphnia. Family is Monidae. Genus is Moina, and the species which we're going to talk about is Moina macrocopa. The other species shown in the picture here is Moina brachiata. The strain which we'll be talking about will be JSK1. So now we're going to go ahead and talk about why we chose Moina as our model organism and why it will be an upcoming model for epigenetic studies. So here in first point, you can see it is sensitive to changes in the water chemistry. To understand this, we're going to see a video. This video was taken by Arunima, and in this video, we can see that a few Moina are orangish red in color and the others are colorless. This shows that it is the Moina is sensitive to water chemistry. So the next point which we're going to focus on is the larger clutch size, which basically means that it produces more progenies in less time. To understand this, we're again going to see a video. This video was taken by Arif, and in this video we can see that a single Moina is giving birth to around 10 to 12 Moinas at once. This shows that it has a larger clutch size. The other points which we are going to talk about is the short life cycle. So one of our cubists has performed an experiment to determine the life cycle of Moina, which we're going to study in the upcoming slides. The next one is Moina is easy to culture. This also we're going to learn how to culture Moina later in the presentation. And the last point is that it has two modes of reproduction, that is asexual as well as sexual, which makes it to be used as a curricular. It is used in a curricular activity to study both the modes of reproduction. Now I would like to hand over the mic to Raj.
My name is Raj and I am from Home Lab, New Delhi. And here we will be looking at the story of Moina, renamed, uh, was renamed from TCTS. So the story dates back to the year 2012 when a water sample from an ecosystem pond in CHM College, Ulhasnagar. As we can see in this picture here, there is an ecosystem pond and the UG system and UG students here are trying to recover some samples from it. And out of curiosity, UG students took out sample from this ecosystem pond and they found water flea-like organism which they thought to be of Daphnia, and they started culturing it as TCDS, that is the chosen Daphnia species. When this culture was passed on to different cube labs across the country, the cube group from Think Lab ANDC, that is in Delhi, led by cubist Kajal, wondered if the water flea was really Daphnia, and they were speculating it to be some Moina species. After consulting national and international network working on crustaceans and Dr. Subhajit Sen from CEBS from Mumbai University, Dr. Sen and the group found out that this water flea is not Daphnia but Moena macrocopa. The characterization of the genome was completed in 2018 and it was submitted to GenBank for verification. And since its acceptance, it is known as Moena macrocopa JSK1, where JSK1 is the strain name and is named after the scientist Dr. Sen and group who characterized it. And in the picture here, we can see that name of the colleagues and Dr. Sen, that is Katti KS, Bhanushali SL, Sen S, and Ramchandani JS. And also below here, we can see the mentioning of Cube as a collaborator in this finding. And this link is also, this page is available the NCBI webpage also.
Hello, Ishita.
Yes. Am I audible?
Yes, yes.
Okay. I'm sorry for.
You got muted, Ishita.
Ishita, your microphone is mute.
Yes. So I'll be explaining about the culturing of Moina. So here, as you can see on the middle hand picture that we have, here we have some water bottles, we have some labels and some empty bottles. Also, we have milk, cotton pads, and the dropper. So the first thing that we need to make sure that we have dechlorinated water. So dechlorinated is water. How do we make it? We just take 250 ml of tap water that you have in your home and you cover it with a cloth and then leave it untouched for 24 hours. What happens after 24 hours is that the chlorine in the water will evaporate and it will not affect the moina after that. So the procedure goes like we use a 500 ml bottle in cube and we put 250 ml of dechlorinated water. Then we go on to collect moina from the fresh water source that we have, like a pond or any standing water thing. And then we put the moina in the 250 ml dechlorinated water bottle and then we feed them with 1 drop of milk every day. Moving on to our next cube list, we have the next step.
Yeah, we'll see a short video on the culturing of Moina. Stay tuned.
So our next is research perspective. So our research question is, what is the effect of hypoxia on Moina and do chromosomal remodeling and epigenetics have a role in it? Chromosomal remodeling and epigenetics will be set out in upcoming slides. Our short-term objective is to study the effect of HDAC inhibitor valproic acid on hypoxia-induced hemoglobin in Moina. Long-term objective is Moina as a model of studying epigenetics, inheritable changes, and observable characteristics as a result through hypoxia-inducible hemoglobin production in the crustaceans as our Moina macrocopa.
Yeah. Good evening, all. I'm Drishtant from Nerul, Navi Mumbai. So while Anshika was introducing us the Moina and what it is all about, she also talked about the life cycle of Moina, right? And when we were making this presentation, we saw that not much information is there on the life cycle of Moina. So I thought that let's perform an experiment to see the actual life cycle of Moina. So I started the setup in my home lab, whose objective, the experiment, sorry, the objective of the experiment was to determine the life cycle of freshwater crustacean Moina macrocopa JSK1, the one which we have in our home labs. And the culture conditions was simply what I took as just these glass vials which are visible in this photo. These are 30 ml glass vials in which I added 25 ml of dechlorinated water, and I fed them with dilutions. But before, so what did I add in these glass vials? For that, I had to do a mini experiment. So what did I do in that mini experiment? I took one gravid female. So this was the gravid female, that is the pregnant Moina. As you can see, this part appears to be bulgy. I harvested this gravid female from my stock culture, and then I added it into another vial and waited it to lay the progenies or give birth to progenies. And I saw that it gave birth to 5 progenies. Then I took all those 5 progenies and then I screened those progenies, those 5 progenies. So what is screening here? Screening is just observing it under the, like, I and the collaborator cubist have formulated one home lab microscopy method by which we use our phones as microscope, using Jugaad methods to simply magnify the organism, the photo of the organism. So screening is that procedure. So I took the progenies from the preliminary vial and I screened them, and I saw that all were juveniles. So I took all those 5 different juveniles and added them in 5 different vials, the ones which we saw in this slide. So each of the 5 juveniles was added in these vials, and then I tracked the life cycle, like I tracked the growth over a period of 4 days. So what was the end point of this experiment? That life cycle means, so I waited for the life cycle to get completed. So life cycle means from birth till the time they get matured and they are able to give rise to their own progenies. So the method that I used was very simple, economical, basic, and yet home lab based. So that is what the motive of CUBE is, to popularize home lab based research, especially in the lockdown times, the pandemic times. So here are the observations. I'll come to the observations later. So here you can see one graph, and the x-axis of this plot represents the time in hours post-birth, and y-axis represents the size of the organisms, that is the 5 juveniles. I averaged all the sizes of the 5 juveniles into one single number, and this is the plot that I got. So you can see one dotted line that is going upwards. So that is the trend line that shows that as the time passes, time in hours passes, the size of Moina increases. So this is sizing of Moina as a function of growth. But here are some important break points that I came across. That after 64 hours post birth, at 64 hours post birth, I can see, like, I was able to see 2 Moina that were gravid, that were pregnant. That means they had eggs in their brood pouches. So eggs seen in vivo at 64 hours post birth. And at the next observation time period, I saw that the progenies had been given out. So let's see how were the progenies looking like. So you can see this was the Moina that I added in vial J1. And you can see that this Moina has a size of around 400 micrometer or 0.4 micrometer. And similarly, I have tracked the growth over the time intervals. And as you can see, I was referring to 64 hours post-birth. So here we can see that there are some eggs here. I have also measured the size of the egg. It was roughly around 134 micrometers, or like around 0.1 millimeters. And the next day we can see that this is gravid, still gravid, and other progenies have been given out. So this was the experiment, and the size that I could make out, the average size was ranging from approximately 400 micrometers to 1.1 millimeter, or 1100 micrometers. Over to you, Saida.
So in Hydra presentation, what we saw that okay, Moina is attracted to light, and even before that we talked about that okay, Moina is attracted to light. So I really wanted to check whether is it just a random observation or Moina is actually attracted to light. So for that, during the discussion, we came up with the experimental design that I had a glass tank handy. So we can put a light source at one end, fill the water, and beneath the glass tank we can keep a black sheet on which we made grid lines, basically making it easier for us to count Moina in one row of column, to see whether Moina is attracted to light and in how much time. So can we see a video for it, Vishna? So this is a video showing the actual experimental part where we were actually keeping the light is kept at this end and the opposite end is also illuminated. It's not that it's completely though there is a gradient form, as in there would be much more light here where the source of light is, and the intensity is decreasing while taking video to see the number of Moinas that we take. And so the total number of Moina that was taken was 80 Moinas, and within 6 minutes.
Sada, there is an overlapping sound as well in the video.
The number of Moinas accumulated over there.
I think you should mute the audio from the video.
Sorry, mute the
Yes, so if you are explaining, just mute that sound because it's confusing otherwise.
Rahul, you can do it from your side. Like everyone has to do it from their respective sides, because this is a YouTube video, right? So just click on your side and click on mute. Yeah.
Yes. Okay. So everyone just mute the video and just listen to Sada's explanation.
I'll start the video over again.
That I have
so this you can see that the light is kept at the opposite end. The whole tank is illuminated. Of course, there is a gradient form. And so within 6 minutes, with the total number of 60 Moinas, 80 Moinas, 50 Moinas actually gathered within 6 minutes at the light source. So that did mean that okay, they are attracted to light. After discussing this experiment in the chat shala, there was a question arose that okay, is the attraction of Moina towards light is just for the white light, or is it specific to a particular wavelength of light? So that is yet to be discussed and sort of form an experimental design for it. And so here we can see the black paper that was kept underneath with white grid lines to basically
divided into 15
count
equal
the
parts.
number of Moina, so that it
And
gets
this is just a tank
easy
with,
for us to count the number of
a little
Moinas.
less than 2 liters
So,
of water. And
yeah, there are some
170
things that we
Moinas in it.
would
We
want
have
to
counted
develop with this experiment,
taken
as
an
in
average
doing
as
it with less sample size and less number of Moinas to
in
actually
the,
track the number of Moinas, because it
box
sort of
that
like
is
becomes difficult
nearest
to
to the light
count the
source,
number of Moinas.
we have counted
Then
more than
trying it
50
with
Moinas,
trying it in
which
bottle,
is
and then
great
also
news. We will
trying with different wavelengths of light. That
just
is
explain the
our
rest
further
in some
objective
time.
with this experiment. So if I was to conclude, the conclusion would be yes, it is sort of Moinas are attracted to light, no doubt. But the question is still there, that whether it is Just the white light, or are they attracted to a particular wavelength?
Okay, so again, we move on to a million Daphnia project. So this project was undertaken around half a decade ago when Moina macrocopa was still known as TCDS. And in this a million Daphnia project, school cubists tried to culture a million of Daphnia, are known at that time. So the objective of the project was to culture 1 million Daphnia using the cube method of culturing. During this, something unusual happened. In one of the culture bottles, Moina appeared to be colored instead of their normal color, that is, transparent white. When the school cubists were asked about this, they hesitantly said that they don't know. But later, one of them said that a few extra drops of milk had been mistakenly added to the bottles, and this was a breaking news. Indeed, it was a goof up, but it was a great result. Since then, PCDS, now called Moina macrocopa, has been studied as a model with research question being accidentally discovered. The question was taken up by many other cubists to identify the reason why did the TCDS turn red. And after many brainstorming sessions, it was identified that the lack of oxygen makes TCDS, the so-called Moina macrocopa now, turns red.
In the earlier slides, we saw a video where we saw that the moina were appearing orangish red. And just now it was talked of that oxygen is actually turning them red. So in the normal conditions, if you look at this picture on the slide, the normal condition, the moina appears transparent. And what do I mean by normal conditions? I mean that the milk is normal, that is 1 drop of milk, and thus the nutrients are normal, and the bacterial growth is also normal, and oxygen is available. So in such a situation, the moina is normal. So what is normoxia? Coming to normoxia, we have to look into the molecular system. Could you please scroll down? So there is the histone protein in the DNA, and the DNA is tightly coiled around it in a normal condition because the DNA is negatively charged and the histone protein is positively charged. In case of hypoxia, hypoxia refers to hypo and oxia. Hypo means less and oxia means oxygen. Similarly, normoxia was normal oxygen. In such a situation, HAT, that is histone acetyl transferase, attaches to histone protein, and histone acetylase transferase, that is HAT, is positively charged, and this it is negatively charged, and this causes the negatively charged DNA to get repelled. This causes uncoiling, and earlier, out of 4, only 1 hemoglobin gene was expressing. But in such a situation, all the 4 hemoglobin genes are expressing and they are being transcribed. This causes more hemoglobin to be produced, and the moina in turn starts looking orangish red, as you can see in this situation. The question might arise that what is causing this less availability of oxygen if we add 4 drops of milk. So if we add 4 drops of milk, we are in turn giving more food to the bacteria. So the number of bacteria are increasing and they are consuming more oxygen, in turn leaving less oxygen for the moina. So now the question might arise, okay, this is okay, but how do cells come to know that there's less oxygen? To answer this question, I hand this over to Rachel.
Yes, Kananika. Before coming to the question, I must say that this meme summarized the previous slide, where the upper half represents normaxic condition and lower half represent the hypoxic condition. And Akshay Kumar is the cell's nucleus. So next slide, please. Yes. So the question was, how do cells come to know that there's a less oxygen? The 2019 Nobel Prize winners, William Klein, Peter Ratcliffe, and Greg Semanza, have answered this question. So let's have a look into their study. But before, let's get introduced to one protein, that is HIF. HIF is hypoxia inducible factor. that has a big role here. So HIF is a protein that is always synthesized in the cell, and it binds to the specific gene that and activates them. But in the presence of oxygen, that is normaxia, it is degraded. That means that the gene won't get activated. On the other hand, in low oxygen condition, it means the hypoxia, HIF goes into the nucleus and does its job for activating the gene by binding to the specific DNA sequence known as HRE, that is hypoxia response element. Now, in the whole story, the gene that we are talking about are hemoglobin gene. So this is connected to the epigenetic. So why we are saying it epigenetic? That because the epigenetic is the study in which phenotype changes, but there is no alteration in DNA sequence. Means there will be no change in genotype. So this is because of the external factor that affects the phenotype and genotype. So here in this case of Moina, it appears red in color when we add extra drop of milk. So let's get to know more about this from Man.
Move further, we must be knowing about the further plans. And so, as to mention them, I would say that our further plans include the to check the response of Moina towards the hypoxia and the normaxia water, that is the low oxygen condition and the normal oxygen condition. Other than these, we want to check the effect of valproic acid on hypoxia induced hemoglobin in using, of course, the Moina. In phototaxis assay, we are recording and studying the phototaxis nature of Moina, that is the movement of Moina towards the light, and extending this very experiment, we want to record and compare the time taken by Moina to reach towards the light source, and we are calling this as motility assay. One question that we cubists often face during the chatshala discussion is that we are always questioned about the dissolved oxygen in the culture water that we are using to culture Moina. So we are also planning to estimate the dissolved oxygen levels in the normal water and also the oxygen deprived or hypoxic culture media.
Okay, comparison of bacteria and Moina. As you can see on the left hand side, there is the bacteria slide, bacteria curve, growth curve, and we have the Moina curve. So at first in bacteria, we see that there is a log phase. So in here, we can see it is not from zero because initially there are bacteria there, and then it goes up, and it goes up to an exponential phase. The log phase right? And then it comes to a halt. The division, the growth stops, and we come to the stationary. And then there is a sudden decrease in number. So there is this death of the bacteria. The similar trend is also seen in Moina. So here we have the log phase, then the exponential phase where the Moinas grow a lot, and then there is a slight dip. But then again, it starts growing because we're feeding them with one drop of milk every day. And then again, if we go on to this stationary phase of growth, and then there is this sharp drop in the growth, right? So this is where the moinas they stop growing at all in the culture. So they decrease, they die in the culture.
If I were to summarize what Ishita said, there was some background noise from her side. I'll do it in a nutshell. So on the left, we can see there's a bacterial growth curve. On the right, we have the Moina growth curve. So how do we obtain the Moina growth curve? So this growth curve signifies, so the lag phase till the death phase signifies a whole like 1 month situation. So this takes around 1 month. That is 28 to 30 days. So from the moment we add some Moina into the culture medium, that is dechlorinated water and milk, drops of milk, till there is no Moina left, this growth curve is on. So as Ishita said that there are some initial Moina, that is why the lag phase isn't starting from 0. We are starting it from the x-axis. And then the Moina will take some time to get accommodated to the conditions that are there in the culture medium, and then as we saw in the life cycle experiment, they will start to reproduce after 2 to 3 days, and so on. It will continue, and this will give rise to an exponential phase or a log phase similar to the bacterial growth curve. Just ignore this slight dip because there are this slight dip must have been due to some like slight decrease in number, so that's all right. This can be ignored. Then the stationary phase goes on till the 14th day, and then the exponential phase goes on till the 14th day, and then from 14th to 18th day we have the stationary phase where the number of Moina that are being born is equal to the number of Moina that are dying. That is why we see a stationary phase. There is no net change, not much of a change is seen in the number. And then after 18, so in the 18 to 28 days, the death phase happens. But here it is shown to be a sharp drop. It is not a sharp drop. It goes down gradually as it does in the death phase. If we move on to the next slide, we saw that I had performed some experiments. Saida had performed, and Saida had performed the phototoxic assay, and we are also planning to do a lot of experiments. We have it in the basket. But where do we actually store all this information of the experiments? We need some log. So in the offline times, we used to have our logbooks and we used to discuss it regularly in the causeries and all. Now we have the Chartshala. But where do we actually just archive all our data? So we have online home lab journals for that in the form of the STEM chat games, our own platform produced by bring forward by HBCSC, TIFR, and it is very similar. You all can just log on to this website metastudio.org, and here you will be able to read posts out that are made for all the model organisms that we have in Cube, almost all the model organisms, be it Moina, be it fruit flies and Hydra and rotifer, like that. And it is similar to our logbooks. Like we can write our updates on that, and it is also like an analog to WhatsApp, because on WhatsApp what happens is we are having some discussions, but those discussions are not being archived unless and until we have it store it in some different repository. But here the topics get saved and we can have a look at it anytime by just searching the topics through names. And it is online and multi-user friendly. You can have a look without even logging in here, so anyone can have a look. You can also write your own blogs on the topic that you feel is relevant to Cube, or not only Cube but STEM as well, like science, technology, engineering, and mathematics. Here you can see I have added Google Sheets, Google Slides, and Google Documents. These are also very essential applications that are used in archiving the photos and videos that we usually take of the model organism. And we can write our design of the experiment. The hypothesis can be written in Google Docs and it can be stored, and we can share it with our collaborators as well. In the previous slides, we saw that the Moina So the life cycle assay of Moina was shown on a slide. So that was archived on Google Slides, and the daily count of Moina is written on an Excel sheet, which is the Google Sheets. And all of this can be shared with our collaborators by just entering their email IDs. So this is how simple it is. So we should just move on the transition to open logbooks. Yeah. Over to you, Arunama.
So we have discussed about culturing of Moina. Now we can look how we manage to culture Moina in different cube home labs. So here we can see Man's home lab in Ranchi, including culture cup, Moina, stock of distilled water. And next one is Shrudiga's home lab in Palakkad and cube lab of Arunima in Kozhikode and Ashwati's cube home lab in Thrissur, Sara's in Mumbai, Drishtant Nerul home lab. These are the home labs for culturing the Moina. And we can see how the collaborators are maintaining Moina in home labs.
So during the Chatshala discussion that happened every day from 5:30 to 9:30 onwards, we came across several concepts regarding the Moina model system. Like
Yes,
like ecosystem, yeah, like the ecosystem, and we also learned that Moina is very much related to other model systems that the cubists are working upon, like the mosquito, butterfly, fruit fly, prawn, crabs, cockroaches, etc. And during that, we also learned to develop the methodology to do the experiments and to be consistent on that. So during this period, the lockdown period, we have been maintaining the home lab cultures since since around 2 years, and we also learned to document them like in the open book what Tristant had mentioned. And we also developed the mobile microscopy through which a very tiny organism like Moina that no layman would have thought that they could document. So this has been achieved by Dristant and the collaborators. So these are the things that we have achieved during the home lab culturing and maintenance of Moina. And our collaborators are not from just one place. We are distributed throughout the India. We are distributed. We are from in the south. We are from Mumbai, Kozhikode, and from the east we are from West Bengal. Uh, East we have from Ishita from Assam, and in the north we have cubists from Delhi, Jharkhand, and also from the southmost we have Kerala, cubists from Kerala as well. And these are the discussions that were going on for this presentation, and these are our collaborators who have taken part in discussions and helped to prepare our presentation.
So now we talk about how is Moina helping us in understanding our curriculum better. We'll start off with developmental biology. Start top most of us. So we are studying life cycle, we are studying reproduction, and reproduction also of both types, asexual as well as sexual. Then the biochemistry of graph, the growth curve, the studies that Drishtan did. Then microscopy. When we talk about phone microscopy, we are understanding the optics of it, resolutions, how to take a picture, how to take a better picture. Then with the phototaxic assay, as in Moina being attracted to light, we are talking about different wavelengths of light, sort of connected with physics. Uh, then biotechnological techniques like PCR, PAGE, electrophoresis can be understood. Biochemistry. So when we were talking about oxygen level, we could understand the dissolved oxygen level by using BOD. That comes under our curriculum and in biochemistry. Can we go to the item? Yeah. So again, taxonomy is a major part. What Anshika was talking about, how different model systems are connected to each other. Then research methodology. Cube always stresses on control, sampling, the experimental design, forming a hypothesis. Then genetics. When we talk about Moina turning red, there is gene expression, there is histone remodeling involved that comes under molecular biology and genetics. Epigenetics, chromosomal modification, the acetylation, deacetylation that we were talking about of Moina turning red. Then gene regulation. So we can use HDAC inhibitor to sort of understand whether the process that we are hypothesizing, the chromosomal remodeling, actually takes place on. Then of course it is connected to Nobel Prize winning work of 2019, and of course microbiology, we can't forget, because Moina feeds on bacteria. So many, it's a one model system. Through one model system we can understand so many different aspects of our curriculum. Moina is a prime example.
Okay, so Zara here, I'll be talking about goof ups. So, goof ups are those simple mistakes we've made and we have celebrated it and we have learned a lot from our goof ups. So, the first one is lack of replicates. So, when we start making cultures in the start, we didn't have replicates, and when our suppose our Moina culture dies, so we wouldn't have another culture to save it. So, we start making replicates to save our culture. So the second is using colored bottle. So over here we have always seen that we could able to capture the moinas because they are plastic, like they are like non-colored, like green color is not used, or suppose any other color which is not supporting the moina, like we cannot see the moina's body. So here we are always using those distillery or water bottles, which is very helpful for us to take the pictures. And the third one is we failed to cover, and so the fruit fly larvae ended up inside. So yes, cotton plugs are very much important, as we even if we didn't notice, and suppose the fruit fly sits on the rim of the bottle, and the larvae might go inside. So yes, the culture might not be as helpful as it was before. So the fourth one was performing experiments without discussing the design. And yes, the design is very much important, as because we can it can lead to the change in life cycle of moina. So designing culturing the moina is very important. And lack of controls in the experiment design. And so here, if we do some experiment and we are having no controls to see that, so how are we going to compare? So this is a big question. So yes, controls are very much important in culturing moinas. So the sixth is lack of daily updates. And over here, like Moina, they have a different amount of population increasing or decreasing every day. So our daily updates are important, as we can see the number of Moinas increasing or decreasing, and we can get to know to other people also that how it depends on the life cycle or lifespan of Moinas. And the seventh and last is adding excess amount of milk to the tank, leading to hypoxic environment. So yes, we when we had a Moina tank in our college, so we added a lot, like a goof up that we did was we added more amount of milk, and what we got, that we got a perfect culture of hypoxic, and we had around 300 moinas. 300 plus in fact, in our Moina tank in just 2 or 3 days. So yes, we had a perfect amount of hypoxia environment we needed. So these are the group ups we did and we learned a lot from it. Thank you.
So recent publications, by the way, let's take some time and read the people who have actually worked on this. So this was a study on cost-effective DNA isolating strategy for crustaceans. And if you read the author, if you just see the name of the authors, they are the same people of the same JSK people who had actually identified Moina macrocopa JSK1 sequence then. Sheetal Bhanushali, Kritika, Jyoti, and Subhojit Singh. So these 4 people had actually found sequence JSK1, and that's how we got our Moina that we currently have, Moina macrocopa JSK1. This particular study actually focuses on using a cost-effective method to isolate DNA using
Hello, Saida.
Sada, are you there?
hello, can you hear me?
Yes, yes.
So okay, I don't know till which point you could hear me, so I could continue from there.
The cost-effective DNA you were talking about.
So so the cost-effective methodology for DNA extraction for these crustacean, and they used actually Moina macrocopa JSK1. They used a porcelain powder-based abrasion. They avoiding like nitrogen handling, because then usually for DNA extraction use nitrogen, liquid nitrogen, to freeze the Moina. So with porcelain powder, we are minimizing the cost. And also the results that they got were actually better yields as per analysis. They got better quality of DNA. DNA quality was good for restriction, digestion, ligation, and PCR, the work that they have done. So it seemed to, it's sort of a jugaad method what Cube believes in. We can sort of replace things with cost effective things, low cost laboratory setups like we've seen home labs. So similar to that, now these people, the JSK people, are actually working on isolating DNA using porcelain powder.
Yes, I would like to thank Arunan sir, GN sir, Subhojit sir, and the Cube family, and also our family members to support us, for supporting us all over the journey that our home lab has, like in developing our home lab and like carrying out the home lab based research and constructively criticizing at each and every step so that mistakes could be avoided, and at the same time they should not be like being a hindrance in the experimental designs when we actually try to perform that, and it will be some sort of a life lesson that we will learn. So thank you everyone for all your support. Yeah, this is the end of the Moina presentation, Rahul bhai. Thank you everyone.
Okay, so that was a very good presentation, and I really appreciate that you thank your family members, because I understand and appreciate how difficult it would have been for them in the first few weeks or days or months even, because you keep a bottle full of water in your home, and anyone can just come who's thirsty looking for water. So the first few days only goes in understanding that, oh, there is now a lab home and we have to be careful about what we should drink and what we should not, right? So it was one thing that we encountered during our Think Lab when we were establishing Think Lab, DC College, and thankful to the principal and Dr. Savitri Singh. So I can understand that family members have part of these kind of and research. But what a journey for Moina, as I said in the beginning, starting from being a food to some other model system to addressing questions which go deeper into the new level, showing that there are genes, upregulation, epigenetics in play, and a lot of and cubists making their way into serious published research. Like that is amazing. I think all of you, past, present, and people who are going to join the Moina group deserve a round of applause. So let's just quickly go to some questions that were asked by our audience. So one thing was in the beginning, you said that you collected moinas from freshwater sources. But how do you collect just the moinas is the question. Like there are lots of microorganisms in the water body. So how do you make a moina culture to begin with? If anyone from the group would like to shed light on this.
Yes, yes. So initially we saw the video of Rachel harvesting some moina from a pond, right? So so what we have to do is, so for example, if I want to tell someone to collect, or if I myself go to collect some water sample, then I'll take 2 water bottles with me, two transparent water bottles, let's say 500 ml bottles or 1 liter bottles, and I'll just dip it in the water body, and whatever I have in the bottles is my sample. Now I'll take that to my home, and the process of separation starts here. So once I let the sample settle, then I'll flash some light. I'll flash some light on the walls of the bottle, and I'll see that whether there are some water flea like or organisms that are seen in the culture. If I see something like that, then I know that they'll get attracted to light. So I need to keep my dropper ready. I'll just dip the dropper inside and take those organisms inside. So this is how I'll be separating the organisms from the rest of the organism, the water free from rest of the organism. And even if we just keep the water fleas in that culture only, that won't make any difference, because in the natural conditions they are anyways living with their other mates, right? So
yeah, so basically you are using the phototactic movement to sort them out from other fellow neighbors. Okay. And in the beginning you talked the group talked about
I have a question.
yes.
There will be a lot of water fleas in the water besides moina, and they will also get attracted towards light. So how will you separate moina from that?
Yes, and exactly that was the question I was also going to ask, that how do you actually culture moina and not daphnias, which we were actually confused about in the beginning. The journey started from there, right? So other water fleas can also.
Yeah, so I think here we'll have to follow the top-down approach. That means we have a general culture of water fleas, let's say. Then we will have to like isolate one water flea at each time, and then if we have some jugaad methods to just magnify their image, then see that which organisms are those. Or even if we don't have such jugaad methods, what we can simply do is we can take one water flea and then add it in a separate culture and start a new culture out of that one single water flea. That means that culture will be of that single organism only. That means it will be a single line culture. And the same can be done for other water fleas, and then we can do a pointed study on that particular organism which is there in that bottle. And obviously here too we should have replicates.
Yes. Okay. That actually I think that's fair enough. And in the beginning, while talking about the culture, the word was used. The word was used that water chemistry affects the moina. Now, what is this water chemistry? Because the first few questions was on just this thing, like what is all hidden inside this water chemistry? What is water chemistry you are talking about?
See, the thing later, in the later part of the discussion, in the later slides, we did talk about moina turning red. and that too was because of adding more drops of milk. I guess that was sort of intended. The term intended was the
The water
change.
chemistry is other organisms growing along with the moina and the pH and what you discussed in later slides, hypoxic condition and everything, right? So I think when you used the word prematurely water chemistry, everyone just was confused. Okay, but that's fair enough. Okay. Also, Dishant, when he was explaining, so this is one of my questions. He explained that graph where you were comparing moina with bacteria, and said that the time. So in the beginning, for the moina, the time taken was for the accommodation uh, in its environment, in the environment. Can we have that slide? Yes. So before the exponential phase comes, the lag phase, that which the reason you said was because of moinas accommodating to the environment. Could it be simply the time that they take to reach reproductive maturity before they start reproducing?
Yes, that can be because see the initial moina that we are adding it like we do not know which developmental stage they are in, right? So we are adding a general like any general number of moina, like let's say we have 5 moina. In that 5 moina, 2 of them can be gravid, one can be juvenile, and the other one also can be gravid. We do not know, so it depends on the number of
yeah,
number and the developmental stage of the moina that are being added.
Yes, and one another question, that why are they dying at the phase? Like you said that the death is equal to the new moinas being born, so that's why there's a stationary phase. But what is their lifespan? Are they dying because of the lifespan? And what in that sense, people ask what is the lifespan of a moina?
Yes, 12 to 14 days in the culture that we are having. Like we culture using 250 ml of dechlorinated water and add 1 drop of milk every alternate days. So in that culture, the average lifespan is 12 to 14 days. So this at the stationary phase, so the stationary phase is reached around the 14th to 18th day. So I think life cycle, the end of the life cycle is responsible. Sorry, end of the lifespan is responsible.
It would also have been great if you would have marked the number of days, or at least one box, signifies how many days or something like that. Okay, but it was a presentation and a good journey for Moina, and it showed that what model system it is. And thanks to the whole Moina group for this very detailed presentation and a lot of work that has been done. And we look forward for a lot of other answers coming in from the Moina group in the cube. So a round of applause for the Moina group. And quickly, let's move to the fourth model system that we are going to discuss today. So going from moinas to, we are going to microbes and rotifers now. So rotifers are they have a very long history, and they were first kind of described or talked about in the early 17th century or the late 17th century or the early 18th century, and also has been described by the father of microbiology, Anton von Leeuwenhoek himself. So what is so special about these rotifers and other microbes that cubists have taken up this also as a model system, and what are the questions that they are asking this system? So let's just go to the rotifer and microbe group. So over to you. So I would just like to introduce the group members: Saida, Deepak, Aditya, Sita Lakshmi, Navneel, and Soumyadeep. So please, before you start, tell us where your cube home lab is. Okay. So over to you, the rotifer group.
Hello. My name is Deepak Sharma. I am from Elphinstone College from Mumbai.
Hello, I am Shaumudik from Kolkata, West Bengal. Acha, like, should we just one by one introduce and then start?
You can just whoever starts speaking can just introduce themselves, the name, and where are you from. So
Okay,
just
then
continue.
Deepak will take over and start.
Yes, Deepak.
Hello. So uh, uh, we talk about biodiversity at our doorstep. So हमारे घर के आसपास में जो मैन होल्स होते जो लीड्स होते हैं मैन होल के, वहाँ पे बहुत सारे ऑर्गेनिजम्स प्रेजेंट होते हैं जो कि बायोडाइवर्सिटी में हेल्प करते हैं. सो हम उन ऑर्गेनिजम्स के बारे में स्टडी करेंगे एंड उनमें कौन कौन से ऑर्गेनिजम्स प्रेजेंट होते हैं उसके बारे में हम स्टडी करेंगे. लाइक इसे हम लोग एक और नाम से जानते हैं, पैगालापोस, which those are related to the Galapagos Island. So, like Charles Darwin survey in Galapagos Island, and उन्होंने देखा कि जो फिंचेस है उनके बीक्स के साइज डिफरेंट डिफरेंट आइलैंड पे उनके बीक्स के साइज डिफरेंट थे. तो उन्होंने सोचा कि ये कैसे पॉसिबल हुआ. तो उन्होंने इसके बारे में स्टडी किया, जिसकी वजह से वहाँ पे डिफरेंट एनवायरनमेंट होने की वजह से, मतलब डिफरेंट फूड होने की वजह से उनका जो लाइफस्टाइल था बीक्स के साइज से उसमें डिफरेंट आया. तो इसी तरीके से हम गैलापागोस आइलैंड से पैगालापोस रिलेट किए. लाइक जैसे दो मैन होल लीड्स है. सो वन मैन होल लीड्स आर प्रेजेंट इन shadow of the tree and another is a present in संदेश वेयर एक जहाँ पे संदेश पड़ता है. तो हम इसके बीच में हम मे भी देख सकते हैं कि जो ट्री के शैडो में है उनका जो एनवायरनमेंट होगा, उनका जो चेंजेस होगा वो डिफरेंट हो सकता है. और जो सन ड्राई में है, जो संदेश में है, उनका बिहेवियर चेंज हो सकता है. सो Galapagos Island is related to Galapagos Island.
Okay, I'm Shomodi, Cube collaborator from West Bengal, Kolkata. I'll take over. So after learning about Pagla Post, that are the manhole leads which contain a lot of biodiversity, like a lot of microorganisms. So I was excited and I went to a venture to find some. And I just went out of my house by building, and I saw some dirty places. Like I looked after for them, and I saw like in between the houses there are some like things lying there, and there is dirt, and there are some drainage system between the houses, which contains a lot of organisms. And also found some puddles in my locality, so I know that I could find many organisms over there. To the next slide. So can we go to the next slide? So yeah, I found something very interesting. I did not find a manhole lid. Can I go to the previous slide, please? Okay, I did not find a manhole lid, but I found where ice cream cups that had accumulated rainwater and some kind of stuffs, and plastic glasses lying around which has got water accumulated and organisms growing inside them. So I call this kind of mobile biodiversity hotspots, and I brought one of them to my home lab. And since I was privileged to have a microscope in my home lab, I took a sample of that and looked under a microscope, and I found a lot of microorganisms under that. And here what you see is a microorganism that intrigued me. This organism is called a rotifer, and this have some interesting property. Like it has rotors in front of their mouth, and this rotors help this organism to like suck in water inside them, and they filter out the water and feed bacteria and plant materials on. And since they have rotors, like the organisms which have rotors are classified under the phylum Rotifer. These are invertebrates, multicellular organism under kingdom Animalia, and This kind of organism with rotors in front of their mouth are classified under the whole phylum Rotifera. So Sai will take over.
So why are we interested in these particular rotifers or any organisms that actually have rotating wheel? We are specifically, if I talk about it, we are specifically interested in Deloitte rotifer. Deloitte is a class of rotifers that reproduces asexually, and there is no male in it. There are only females in this particular rotifers, in this particular class of rotifers, and there's a lot of diversity seen, not just in shape and size of rotifers, but also like the diversity is so much so that some of them are oviparous and some of them are viviparous. So some are laying eggs and some are giving direct birth. So how is this diversity seen? Of course, we know that it could be due to mutation. It could be an answer to the question that we are asking. Or if but still so much diversity, other than mutation, what could be the possibility? Because it is sort of cloning themselves. Because there's only one, there's only mothers. The mothers would sort of clone because mutation may play a role. So there are identical offsprings. So such a diversity form is a question. So when we actually look into the literature, we found that okay, there could be horizontal gene transfer. I know it's a big term, but we do come across this in our curriculum with respect to bacterias and antibiotic resistance in bacteria. So if I was to simplify, I can take example of humans. Humans do vertical gene transfer, where the gene transfer is between the same genus. Here, there could be gene transfer between two organisms that are completely different and they don't belong to the same genus. Like transfer of gene from rotifer, from this Deloitte rotifer to bacteria. Sorry, bacteria to Deloitte rotifer, fungi to Deloitte rotifer, then plants to Deloitte rotifers. And by the way, the name highlighted, Matthew Michelson, is on the same Michelson that we come across in our +2 for Michelson and Stahl experiment. So his group of researchers, when actually looked into rotifers, they found that there are several genes to be have originated from bacteria, fungi, and plant, depending on the environment in which rotifers were found. So they hypothesized that it could be horizontal gene transfer. Maybe they are taking up genes from the environment, though they did not conclude that it could be a horizontal. It is a horizontal gene transfer. They have not concluded it, but this is their hypothesis. So, well, this is another article that talks about the identification of the rotifer. So basically, if you go back to the video, here the term written is for Deloitte actually is the retractable foot. So if you go back to the video, the tail actually retracts, and it retracts similar to what is seen, the retraction that is seen in telescope. That's why the article referred it as telescopically retractable foot. So that could be one key features to identify Deloitte. These are some evidences of Deloitte reproducing asexually. This is a particular rotifer, Edetic vega, that in which they found that there is asexual reproduction. So how do we actually go about how do we actually go about studying this horizontal gene transfer? How do we prove or disprove this? If they are taking up gene from the environment, like what Matthew Michelson suggested, then and they feed, by the way, Deloitte rotifer feeds on bacteria. The rotifers in general feed on bacteria. So if we give an environment rich in bacteria, say bioluminescent bacteria, and if the bioluminescent gene gets transferred in rotifer, then the rotifer would glow. And if that if that is passed on to the next generation, if that glowing the luminescent gene is passed to next generation, we can say, okay, HGT has taken place or not. We can sort of prove or disprove the hypothesis depending on the results that we get. But okay, before lockdown, we could culture bioluminescent bacteria in labs using agar medium. But how do we culture it at home?
Yes. So I will take over. So keep looking for alternative media to grow some microorganisms in our home labs. Since we do not have very sophisticated techniques or objects to do equipments to culture bacteria in our home labs, we started using some very household common things. And once the one picture that is on the left shows like organisms growing on a petri plate. That is how it's done in the labs. On it, it's done by Arun sir. It is a very household method where he had inoculated or put some bacteria in S shape on a boiled potato slice where the microorganisms had grown over time. Potato slice was used as an alternative to grow microorganisms in our home labs. To the next slide. Okay. So what we see is I was looking for some media to kind of grow microorganisms, and one of the very common media that I found was coconut. So coconut water seemed to be a very nutritious media, which is naturally available and also very like easily available to us. And what I did was to study the growth of microorganisms, not study, but to find out. So I cut the coconut and took the water in a conical flask, left side top picture. And I saw it's very clear. So to check how clear it is, I put a book behind the container, and I saw that I can see the pages or the alphabets of the book, right, written. And after like 9 hours, I did the same thing, and I found that the water has turned turbid. The coconut water has turned turbid, and I cannot see through it. So, and after 24 hours, it had turned turbid, and there is froth, white froth forming on the top of the of the coconut water. So this shows that organisms had started growing, and they're like very much grow on the coconut water. Similar kind of things are used in our household, like toddy is used like for inflorescence from coconut tree, and those are also used in food preparation, and toddy is used as a beverage also. So it's some kind of fermented inflorescence from coconut tree. And some similar was further carried out by our cube collaborator Aditya. Aditya, you can take.
Yes, thank you. So myself Aditya from Kalyan, Mumbai. So as mentioned rightly by Somyadev, the toddy is the fermented alcoholic beverage prepared by the sap of the coconut inflorescence, that is collect the vessels and then left for fermentation for a few hours, and then it gives rise to the toddy, which is an alcoholic beverage. And similarly, this coconut water that is studied by the Somyadev, fermented coconut water is also traditionally used in adding for addition in the dough of making a bread or sana kind of thing, which is prepared in the southern parts of India and also some parts of Goa for making fluffy, and it is added to enrich the flavors. So why not to give a try by using fermented coconut water? So what I did was simply collecting the coconut water in the bottle. I left the bottle for the fermentation. After few hours, I just dipped this in the picture. As you can see, I just dipped the cotton earbud, which is easily available at home. I just dipped it in the water, as it will absorb the coconut water, and streaked it, or we can say spreaded it on the boiled potato slice. Next, please. In the S-shaped manner, here we can see the container. It was a white plastic box. I put the boiled potato slice there and streaked the this fermented coconut water shaped manner, and I just closed the lid and left it. So these are the results after the 48 hours. We could see some sea colonies, some yellow colored colonies are there, and something is available. So So here we can see that boiled potato slice can be a good media for growth of certain microbes.
No, Navneet, you are not audible.
Okay, so Navneel also tried a similar experiment as shown by Aditya to actually grow microbes at his home lab. So what he basically did was he boiled the potato for 25 minutes, and it was, of course, the shape of the potato was not as intact as it was for Aditya. So if you could see that it is turned a little mushy. So while inoculating, taking a picture of the microbial growth, uh, we couldn't actually. Yeah. Okay. Yes.
Okay, thank you, Said. I'm so sorry for the interruption. For some reason, my mic was not working. Okay, so when I first saw Aditya's experiment in the Chai session, I was extremely inspired. At that point of time, I was actually trying to set up my own home lab here in Kolkata. I am from St. Xavier's College, and I was trying to set up my own home lab, and I was extremely inspired by the way he used a boiled potato to generate microbial growth on the surface. And I took a hint from him after I joined Cube. I also planned that why don't I do the same thing? For that experiment, I was going to replicate his experiment only by taking the fermented coconut water and putting it on a slice of boiled potato and trying to see if I could get the same results as Aditya had. However, when I boiled the potato and I tried to slice it, I couldn't slice it properly at all. I noticed that I had boiled the potato for so long that it had actually become a potato mash and I couldn't slice it anymore. And if you know that the plain surface is extremely important if we wish to see microbial growth properly and if we wish to notice what type of microbes are growing as a whole, the plain surface is extremely necessary. Therefore, when I got a mash of potato, it was very difficult for me to even put the inoculum on the surface in a regular manner. Therefore, at that moment, I realized that. So when I actually did the experiment, I actually confirmed one of the hypotheses of Aditya that yes, potato does support microbial growth because after 24 hours, I did see some amount of microbial growth, and after 9 days, which was actually a goof up because I became sick during that period of time, and the potatoes were left untouched. And after 9 days, there was considerable amount of microbes growing on the surface of the potato, as you can see in the right hand in the right hand most picture where it is written 9 days. And after this particular goof up, we had a discussion on Chatshala to understand why this happened and what we could possibly do about it. Therefore, we came about with a plan that we could possibly do an experiment where we standardize the amount of time we are boiling the potato so that every collaborator who was trying to replicate this experiment, who was trying to use this particular potato as a medium for microbial growth, could boil the potato under the same conditions and have the same surface of growth without worrying that their surface of growth might be affecting their experiment as well. Therefore, if you can please move to the next slide. Thank you. So accordingly, we came about together. We came about with a plan, basically an experimental procedure to standardize the amount of time that we are going to boil the potato. So we boiled the potato. So first, I took 3 potatoes for 3 different temperatures, which is 5 minutes of boiling of the potato, 10-minute boiling of the potato, and 15-minute boiling of the potato, knowing very well that if I breach the 20 to 25 minutes mark, which was my previous experiment, then the potato will become a mash. Therefore, once I decided that, I took an open pan, put water inside it, and put the flame under high heat. It is not simmering but high heat flame. And then I let the potatoes boil in the water for 5 minutes, and then I took out one of the potato. Then after 10 minutes of boiling, I took out another potato, and after 15 minutes, I took out the last potato. After I had taken out all of these three potatoes, I let them cool for 1 hour in separate plastic bags. Once they had cooled down significantly and they were suitable enough to hold in my hand, I used a scalpel which I had previously flamed. Basically, passed it over a fire to make sure that there was no microbes on the surface of the scalpel when I'm slicing the potato. And then I sliced the potato, every single potato, that is the 5 minute, 10 minute, and the 15-minute potato, into regular rectangular slices, and took 3 slices from each potato. On the surface of the potato, I did a very interesting thing. So when I was watching the television, I actually noticed an ad, as well as in the road today also I noticed an ad of this particular drink called Yakult. So what Yakult was claiming, basically, that it was a probiotic drink which had 6.5 billion bacteria included in 60 ml of its bottle, and that was quite the claim. So I decided, why not let check the growth of this particular bacteria on the surface of a potato. So I took the inoculum and then I diluted it a bit. By a bit, I mean I took 1 ml of the inoculum and put it in 10 ml of water. Once I had the inoculum diluted, I took a earbud and put it on the surface of each of the potatoes. So let's move into the next slide. Okay, so this was my experimental setup. Basically, I had a 5-minute row, 10-minute row, and 15-minute row, and 3 replicates in each column. That is 1, 2, and 3. My last row was a control set of potatoes. Why did we need a control? What was the control? That is very important. The control was actually slices of potato, boiled potato, from the same 5-minute potatoes, excepting the fact that there was no bacterial inoculation on the surface of the potato which was made. This was very important because we want a particular organism, as much as possible, a particular organism growing on the surface, so that the growth characteristics of that organism, if it is growing fast or slow, does not affect our observation that the potato is actually influencing the growth, when in reality it is probably the microbe which is growing fast or slow. Therefore, if we got some organisms growing on the control slide, and we saw the same organism growing on the potato slide as well, so we can compare both and say that this is a particular contaminant, and we should not consider the growth of this particular microbe on the surface of the potato. So let's move into the next slide and what I observed. This was my 0-hour pictures, where I have a control, the first replicate, the second replicate, and the third replicate. And after 24 hours, the first thing which I observed was there was a general browning of the surface of the potato. If you can, yes, just stay on this. Oh, yes, right. Yes, thank you. If you can zoom in into the control first, please. The control is on the left-hand side. Yes, thank you. So at 0 hours, the control is completely clean. And even at 24 hours, let's go down a bit, please. Yes, even at 24 hours, there is no perceptible change other than a general browning of the surface. For the first replicate, we notice a slimy, shiny growth. Wait till it comes. Okay, we are nearly there. Okay, thank you. So if we so if we look at the 5-minute replicate, then we'll notice a shiny white, shiny translucent growth on the top left-hand corner of the potato, which is not present in the control. Let's go to the second replicate, where we will notice the same thing actually, but on the top left-hand corner in this case. One important thing to notice is that this is not happening anywhere else in the potato, but at the top corners right now. But in the third case, there was a bit of an aberrant change, when we noticed that there was more growth than the first two cases. This was just an observation which I made, but to note is that the rest of the potato and the majority surface was actually still uncovered by this bacterial growth. If we can move into the 10-minute, the next slide, please. Thank you. So in this case, there was one thing of notice, which I don't know how it affected our experiment, but the potato, even at the 0 hours, was actually showing some amount of browning on its undersurface as well. And if we move into the 10-hour phase, we see that there is a complete blackening of the undersurface of the potato, along with a shiny whitish translucent growth on the surface. Since this was present in the control as well, and if we look into the 5-hour piece as well, we will notice that there is some similarity in the two growths. Therefore, we are sure that this is a contaminant of some sort growing on the surface of the potato. However, the shiny translucent growth, that is definitely bacterial, and this was not present in the control. And if we move into the other potatoes, even though we see the contaminant, however, we also see a comparative growth of the bacterial populations as well. And this was present in the three of the replicates along with the contaminants as well. So if we move into the 15-minute, please. Thank you. So in this case, and this was very interesting, the 15-minute potato did not show any browning at all. It was just as if it was at the 0-hour phase, excepting the fact at 24 hours, if we zoom in into the picture, then we will actually notice that the surface is extremely shiny. There is a complete coverage of a translucent growth throughout the potato. And if we delicately zoom in into the right hand side of the first replicate, we will actually see bumps and irregularities, which are basically the microbial growths on the surface of the potato. And this is how it looks all over the surface as well. So if we move into the second replicate. Yes. So in the second replicate of interest was actually central colony, which was completely differentiated from all of the other 8 replicates which are present in the experiment. This was when I actually tried to pick it up, it was more of like a milkmaid type of substance. It was very sticky, very viscous growth, and this was completely different from the rest of the potato as well. Since it was growing on only this potato, it was most possibly a contaminant. The reason I say most possibly because I did not observe this on the control, and the rest of the potato was once more covered by those undulating masses of translucent whitish bacterial growth, and it was the same for the third mid replicate as well. So this was the end of my experiment, and from this experiment it was quite possible to observe that the 5-mate potato did not support as much growth as the 10-mate potatoes and the 15-mate potatoes. Therefore, we may suggest that the 15-mate potato possibly supports the best growth of microbes without turning into a mash and giving us a plain surface of growth for microorganisms. And this is some sort of standardization which I carried out.
Yeah. By seeing that, okay, potato slice can be used as replacing the agar plate basically to culture microbes is also a general interest, as in we can like what Navneil, Aditya, Soumyadeep did, using kitchen using the things that are available as in kitchen like milk and coconut water to actually identify, to see whether potato slice actually can be used to culture microbes. Similarly, we can sort of use this potato slice for culturing bioluminescent bacteria and probably carry out the experimental design. But the experiment for understanding whether horizontal gene transfer actually takes place in rotifer or not, but that would be a future plan, and we would discuss this in Chatchala and develop it more before doing it. I think the first initial objective would be for rotifer group at least to start of culture rotifer, and also what we have seen with Drishtan's experiment with Moina group and fruit fly group, we can use phone microscopy to sort of see these rotifers and sort of identify it and start culturing it at our home labs. So Abhijit, can you?
Yeah, so we started with rotifers, understanding the mechanism, how it was. So how all this sort of microbial studies that we are doing, experiments that we are conducting, and all of this, how we can connect them to our curriculum. With rotifers, we can understand genetics, and the genetics we can study protein synthesis. And protein synthesis, we start learning about protein synthesis since we are in ninth standard. So we can study about transcription, translation, and translocation, and about DNA and RNA and genes. Then about amino acids we can learn about them. So the sort of things we can understand. Then we have developmental biology. In developmental biology, we can understand about how reproduction occurs. When Saida mentioned about parthenogenesis occurred in rotifers, like they are almost like cloning themselves. So that we can understand with rotifers, with haploid parthenogenesis and then diploid parthenogenesis and stuff like that. And then we comes across molecular biology. In molecular biology, we can understand about horizontal gene transfer. In our college curriculum, we come across those terms, and it is very important to have a context to understand them. So we have conjugation, then transformation, and transduction. So these all stuff we can relate with by having rotifer as a context. Then we have bioluminescent bacteria. So as we said that we need culture, a solid surface media to culture bioluminescent bacteria, to culture, to understand the entity rotifers. Then we need nutrient agar medium or potato slice that we have developed to culture bioluminescent bacteria in that. So that we can understand with. Then we have parallel pose, which is developed, which is an innovation of cube, and through parallel pose we can connect it with Darwin's theory of natural selection, and we can study evolutionary biology. Then in evolutionary biology, we can understand mutation, then variation, then natural selection, and the survival of species. That kind of thing can be studied. And then we have research methodology. We can understand about sampling, and how much replicates we need, and how much control do we need. The literature review, we can go through the previous work which has been done, and how to look for reliable sources. So this sort of things that we can come across while studying rotifers and microbes, and we can connect it with our curriculum. So that is what how we connect it with, and I think now we are open to the questions.
Okay, so another very good presentation by the group. So it was very good to see where we as cubists are trying to develop alternatives of sophisticated research methods like agar plating, and we have our own potatoes now instead to do the microbial research. So I think, and also the development that we have done in the field of microscopy using our own mobile cameras, that is present with everyone these days. Okay, so let's go to some quick questions. So when you were discussing about the microbial growth, would you like to talk about whether questions have come that how would you differentiate the microbial growth? Like when you say microbial growth on a agar plate, obviously very intuitively the first picture that comes to any person's mind who has studied microbiology is that these are bacterial colonies. They look like bacterial colonies, right? So how are you going to differentiate between different microbes? Like it could be fungal colonies or bacterial colonies, like various different microbes growing, right? This was one of the questions. So
Basically, we even in colleges we come across this, like when we culture microbes in using agar plate as well. Then we just don't go for identification using like difficult techniques. Just like that, we sort of use colony characteristics to sort of identify what kind of organism is growing on that on the agar plate. So similarly, even in the potatoes, like the same thing is applied. We can through
Sada, your microphone is mute.
sorry, yeah. So even with potato slice, we can do the same thing through colony characteristics. We can sort of try and understand what kind of colony is. What is it, bacterial? Is it fungal colony?
So in an agar plate, we can actually predefine conditions that will allow certain microbes to grow. Because when we say microbes, I think the question in people's mind came from the point that when we say microbes, it's not something very specific we are talking about. We are talking about everything that we cannot see with the naked eye. We need a microscope for that, right? So what are we looking forward to? Yes, with this innovation.
Yes,
How
we are in
closer
the
we can
very
bring it to a agar culture plate? Yes.
that actually it's very juvenile stage of culturing microbes. We had just started with finding the very common nutritious media that can grow that we can grow microbes on, and I think collaboration in cube we can come up with more specific kind of media. We can process some kind of nutrients so that we can only grow bacteria, or very specifically we can do something for the fun for growing fungi. So yeah, we think we will be doing that in future. And even rotifer culture, we will be developing on that so that we can also culture the rotifers properly. And hence the motto of the experiment is to study horizontal gene transfer thing in the rotifers. So we will be doing that finally.
Yes, and that's good. So that's what I was asking. That what is the road ahead? Like, do we see this potato-based culturing going forward and in some time replacing agar-based culturing for microbes? And specifically, we can figure out what kind of microbes we can grow. That is one thing where you have to go, and that's a great thing, I think. Also, when we talked about culturing microbes, so would you like to talk about what do microbes feed on? Basically, this question is by someone, and it's I think pointed towards the nutrition. So, what are we ensuring to these microbes in terms of nutrition? Do we know about their nutritional requirements already? Like, is there something you would like to say about this?
Uh, Okay, yeah. So microbes, we know it very specifically from the microbes that we had grown, we had grown them in lab. So we know that we need some kind of sugar, that is glucose, and we also had some kind of protein source that help they help getting them nitrogen, phosphorus, all those things. So all these are very much found in the potato slices also, as an abundant. So since these very basic needs are present in the potato slice, they very in general have microbes to grow. So some kind of sugar, some kind of protein source, and some minerals that are present. The
Yes,
microbes will grow.
and one standard question in the public chat was that why are you actually boiling the potatoes? So what do you think will be the difference if you don't boil the potato? And can we do something of this kind on a raw potato slice for that matter? Or is boiling uh, is important for something which is happening? Because we can understand that in context of our agar plates, so that we dissolve it, we boil it, and when they settle down in a jelly like material, which is like a mat or a substrate for microbes to grow on. So what is this
Yes,
boiling
sir.
doing with potato? Yes, Naveen.
So we have some theory about it that we are going to test it in the future experiment. The theory is that the simpler the molecules, like if we break down carbohydrates, as Shomadeep said, sugars, if we break them down, the more broken down it is, the easier it is for the microbes to actually take it inside them. Like when we eat our food, even our food needs to be broken down when it undergoes goes through our digestive system and it undergoes breakdown into smaller molecules. So when we are heating something, it actually leads to the general structural degradation of most molecules which are present inside the potato, and they are usually broken down into simpler substances. Our hypothesis is that the main component of potato, which is actually a carbohydrate called starch, is actually broken down into simpler materials. So in the future we actually wish to
Yes,
uh,
yes.
yes, so in the future we actually wish to do a starch test using iodine and see if this is true. That does boiling actually break down the starch into simpler materials or not. So that is why we are boiling the potatoes.
But then the question arises along the same line that we are here boiling the potatoes for bacteria or to feed them, but the cooked food is not being provided to them in the nature, right? We also see bacterial colonies growing on raw potatoes when potatoes undergo.
Can I?
Yes, Aida.
So, what Batul ma'am was doing yesterday, if you guys remember, yesterday we were discussing about nematodes, and Batul ma'am was actually using a raw potato. She's putting milk in the center, and the nematodes were attracted towards the center. Basically, the idea was the milk will enhance the growth of bacteria. So, what I feel when I think about this boiling of the potato, that even raw potato can be used uh, in general, because there are some microbes that can break down sugar, right? So
Exactly. So because the whole question was that are they depending on us for to boil the potato for them? They must be having some innate mechanisms.
yeah, so even they can be cultured on the raw potato is what I get to know from Batul ma'am's experiment. And other than that, there would be like several things, not just potato would be the only media for microbes to grow. Like
So can there be another reason associated, because Simran has written that aren't you boiling the potatoes for sterilization? Is it the reason for the cubists?
If I can take that question, so the sterilization is a very important step, since if when in lab when we're using agar, we ensure we actually use our autoclave to ensure that it is completely sterilized. So when we are actually boiling the potato, we are doing a sterilization step, but that is not the only aim. That is definitely a sterilization step, but that is not the only aim of boiling it.
Yeah, but the other aim is actually it's not very much required, as Saida said, that you can see growth of microbes or bacteria on raw potato itself. But I think to begin with the raw potato, it will be difficult for you to tell whether the potato already had that bacteria or it was infected, or whether the one you are growing is being cultured, right?
So
Yes, that
it
is
all
absolutely true.
depends on the question that we are asking. If you are asking a question that okay, can potato be used in place of agar, even raw potato can be used. And if you are asking specific questions, like suppose if I'm putting milk or fermented coconut water to see what is there in this coconut water that is fermenting this coconut water, if you want to see that kind of microbial growth, then I think we surely need to boil it for sterilizing, is what I understand.
Yes, like in my case, this particular experiment was actually a continuation of Aditya's experiment where he used a boiled potato. So my intention was to only find out if what was the perfect temperature where we could get a plain surface to actually grow microbes on it.
Okay. Okay. So if I can add a little bit on that, so boiling a potato helps like more nutrients to get to the bacteria. So we had found like on raw potatoes, not so much bacteria is growing as compared to a boiled potato. So we
Something
prefer.
to that, I would say, have you done a comparative study like simultaneously in
Uh,
the same
yeah,
environment?
that's a good thing. We will be doing that, and I will just get the
Yeah, so we
pictures.
can
That
sort
will
of
help.
work on it and then sort of design an experiment to check whether boiling is really needed or not. For the question specifically whether microbes can grow on potato snacks. But if we are asking a specific question that okay, are microbes from the milk or from the coconut water are growing, then for that I think we need to boil. Can we take up other questions around? There are any.
Rahul is not around. So if we do not have any questions, then we can continue with the next model.
Okay then. So the next model system that we have is mosquito model system, and we were actually talking with respect to how is fruit fly or how is moina connected to mosquitoes. So without any further ado, I would ask the mosquito group to join in and would request that each one can please introduce yourself. So the group consists of Meena ma'am, Abhijit, and Gaurangi. Please introduce yourself. Where are you from? Where is your cube lab? And you may proceed.
Uh, Good evening, everybody. This is Hina Mudgal from Cube Kanpur, and we have been working on the mosquito model organism since year 2018, and we have an extensive collaboration all over India with Cube Kochi, Cube Ranchi, and collaborators from Assam, and we have collaboration with other states of like we have people from Mumbai who have just roped in, like Gorangi and Abhijit. And in this model organism mosquito, it's basically it has been assigned as the citizen science project because we've been doing certain home lab activities with school children as well as with college children, and we are working upon different types of surveys, questionnaires being filled, and we are talking of the awareness and spread of information that how this threaded mosquito, which looks like a harmless organism, is spreading diseases. And we have done a series of activity in our home lab, which is the Cube Innovation. That is the celebration of the Cube National Meet. Like we have had 500 consecutive days of the evening Chatshala session. Here we have innovated the home lab concept that the experiments can be done very simply with things readily available at home. We don't need an expensive laboratory to study science. Similarly, with the mosquito model organism also, we did this innovative setup after being motivated by Cube Kochi in the last meet. We set OB traps all over. We had a series of classes of students who had set OB traps this time. We had taken up OB trap setup from August and September. This was the new innovation which we did, and actually we tried to replicate the Cube Kochi group. And here we have also done studies of the breeding places, and all our collaborators who are here will share their experiences with the slides. And we have collected samples of larvae and pupae, and even the mobile microscopy which has evolved. And we have used the mobile in a way that we can make the best out of it without having a microscope at home. And students have shared their pictures, and we have uh, we are going to showcase that. And now please may I see the next slide, please. Now the main objective of the mosquito study is it is a citizen science project, as I already told, and the initiative is of the various cube groups to bring awareness of the vector-borne diseases like dengue, chikungunya, Zika virus, and yellow fever, because these are the diseases which are being, carried by this mosquito. It is a vector, and it is really a dreaded mosquito, and this has to be brought in the common mass. How to check its breeding? So we have been studying on the Aedes species and like Aedes aegypti, albopictus, and recently we have tried and seen Aedes vitatus also. So over to Abhijit and Gorangi.
Okay, so as ma'am said that we are working on the mosquitoes so that there are a lot of diseases spread due to mosquitoes. We all have seen mosquitoes all around biting us and disturbing us. And we also know that mosquitoes spread a lot of diseases. Different mosquitoes spread different diseases. We are working on Aedes mosquitoes especially. We'll tell later how to identify Aedes mosquitoes. So our research question is prediction of types of mosquitoes breeding in an area in a particular month or a season. So we are graphing our mosquitoes according to the locality. We are doing it all over the India, as Hina ma'am is from Kanpur. Sir, you wanted to speak anything, sir?
I guess, Gaurangi, you can continue.
Okay. So we are graphing it according to the months, and we are comparing it with the season cycle. So this is the season cycle. We all know that there is two equinox: March equinox, September equinox, and two solstices in the whole season, that is June solstice, and that is called summer solstice, and then the winter solstice is in December. So we are trying to find that when is the dominance of the mosquitoes, and which type of mosquitoes dominate in the whole year, in which season. So we are doing this season comparison. So we are predicting that more number of mosquitoes, of Aedes mosquitoes, will be more during the rainy season, as we all have heard somewhere in the news and by some teachers and all, that there will be more mosquitoes in the rainy season. So we are trying to so that is our main question, that is that true, that is that mosquitoes are more in this June to September time, that is the rainy season, that is from June solstice to the September equinox. So this we're trying to find it out. Then coming to the mosquito body parts. Now we all have seen mosquitoes. The mosquito belongs to the phylum Arthropoda and class Insecta. It has three basic parts: the head, thorax, and abdomen. As it is characteristic. It is a characteristic feature of of the phylum Arthropoda that it have three basic parts. It is divided into three basic parts. The head region consists of antenna, maxillary pulse, and the proboscis. Mosquito uses its proboscis to suck the blood from our body, and hence we get irritated. Then the thorax region, on the thorax region, the legs and a pair of wings is connected at the thorax region, and then it has the abdomen where it stores the blood and its food, etc. Now moving to the next slide. So now Abhijit will continue this.
Yes, so as Gaurangi mentioned about the body parts of an earthworm, so now let us have a look on the life cycle of mosquitoes. So when we have adult mosquitoes, then the adult mosquitoes lays their eggs in water bodies, which is in lakes, ponds, and the stagnant water area. That part, the places mosquitoes lays their eggs, and then the eggs gets hatched into larvae, and the larvae get converted to an emerging new adult flies, and then the adult mosquito is ready to bite us and disturb us during our this thing. So this is what is about life. So as we were egg in the stagnant water, so there is a high, and it is because in rainy season the amount of the water, like the breeding places, the possible breeding places of mosquitoes increases. So sorry for that. This photo on this first photo was captured by me, and I was just hanging around in my balcony, and I saw this substance which was in my neighbor's. Saw that there is a stagnant water because of rain. Water has got collected at that place, and that can be a possible breeding site for mosquitoes. And this was of this month only, like the first week of September. And then the second photo is of the coconut shell in that the water is collected. And this photo was captured by Kiran, and this photo is from HBCSC campus. At HBCSC campus. So when we talk about that mosquitoes, we found more number of mosquitoes in rainy season. So I think as we were discussing through discussion, we kind of say that in rainy season the possible breeding sites for mosquitoes increases. So such places can be the mosquitoes can lay their eggs there. So the main aim is to spread awareness among people that we should avoid the water getting collected in such spots or places, uh, and that is what the main project is also there. And also, this is a slide of how we can identify the Aedes from the other species, Aedes and non-Aedes. So actually, Aedes mosquito is having white stripes on their body. Like in general, the genus Aedes is have the mosquitoes of the genus Aedes. They have the white stripes in their body, and also we can observe different patterns in the arrangement of this white spots. And we have different species like Pigeon and Rotocactus and the other ones. Then we have Culex or Anopheles mosquito. They are mostly yellowish brown kind of, and they don't have any such white kind of stripes or on their body. So this way we can identify between Aedes and non-Aedes. So this photo was captured by Rahil, and he is a collaborator from QCAM. So, so yeah. So first of all, to predict in which season we get more number of mosquitoes, we need to catch mosquitoes. Because it just in air we cannot count it. Yeah, this is the mosquito. This is the mosquito. So what we do, we try to develop some ways like simple claptrap method. Like this is what. Sorry for that. This is what we develop. Like we are just clapping our hands. When the mosquitoes come to bite us, we just give them a clap like thing. But there is a photo on the top right side. You can see that. So we just clap. But the thing is, we just keep a hollow between both the hands, so that the mosquito ka body won't get squished and broken down. Its leg won't get detached, and that stuff won't happen because we require that for identification between the non. So that is why it is important that we don't crush the mosquitoes only by clap. So we just maintain a little bit of hollow in our clap, and we just trap the mosquitoes with that. Then the other method which we have is OV trap method. So OV trap method is that, so what we do is we just take a cup and we place the ice cream stick and cover it with a cloth, white color cloth. Then we just cover, and then we place this cup in the place where we think that, and the next day or the days we can see that the mosquitoes have laid their eggs on the substrate, that is the ice cream stick covered with the cloth eggs. So with that eggs we can identify which type of mosquito in that area. It is mosquitoes egg. Then if it is white color, then it will be probably of Culex or Anopheles, like that. Developed for cat. So can you go to the next slide, please? Yeah. So this is the hypothesis that we made in 2000. We are working on this
So,
project since 2017.
I will I'll take upon the hypothesis. I'm Gaurangi Dhuri from Cube Mumbai. So we used to work in school days on this hypothesis. So now we have resumed the work. Myself and Abhijit, we are in the same school, and that time during the discussions in the Cube lab, we we found since we hypothesized, we had two hypotheses. We also had one more school working with us on this mosquito project, that is Devnar school, in which there were many students actively participating in this cube work. So from that, one was Ubaid and one I am Gaurangi. So we had two hypotheses. So this hypothesis is of 2017 year, and we hypothesis the total number of mosquitoes over the Aedes mosquitoes. So this blue line in both the graphs is showing the total number of mosquitoes, and this red line is showing the Aedes ratio out of the total number of mosquitoes. So according to my hypothesis, the peak of the total number of mosquitoes would be found in the April-May month, and whereas there would be least total number of mosquitoes in the June, July, August month, that is the rainy season months. But the Aedes ratio would be the highest during this rainy season, that is June to September. Sorry. So and then again the Aedes ratio would start to decrease from October, November, but the total number of mosquitoes would start increasing. So that was my hypothesis. And Ube's hypothesis was that the total mosquitoes would be at its peak during the rainy season, that is from June to September. Sorry. And whereas the Aedes ratio would also be at its highest in the June to September month. That means 90 to 95% of the total mosquitoes would be Aedes mosquitoes. So in both our hypotheses, we have one similarity, that about the Aedes ratio, that we both thought that the Aedes ratio would be at its highest, that is about 90 to 95% would be Aedes during the monsoon season, and then the Aedes mosquito would start decreasing. But in case of dissimilarity, we had the dissimilarity that the total mosquitoes, I thought that the total number of mosquitoes would be most during the means from March to May. Here you can see the peak, and it would reduce in the monsoon season, whereas he thought that there would be a peak of total mosquitoes in the rainy season. So we used this hypothesis, both the hypotheses, to work for a whole year to find what is exactly the scenario, or is it something else. This is our work. These are the mosquitoes caught by me by the claptrap method. As Abhijit said, the claptrap method. We made hollow in our hand and we trapped the mosquitoes. We didn't. We have to take care that we are not means going somewhere to just catch mosquitoes, or keep windows and all open to invite the mosquitoes, come, I'll catch you, come, I'll catch you. No, we are not going to do that. It is just we are trapping the we are just collecting the mosquitoes which we usually kill, but we just throw out. So similarly, these are the mosquitoes of me, caught by me. So just collected it, and then we kept it under on a black paper. As Abhijit said that Aedes have the white stripes present on their body, that is on their legs and their abdominal region. So that to identify it easily, we kept it on a black background, as we all know that it would be quite easy to identify them. So this is a black paper on which we kept I kept all the mosquitoes, and all of them are Aedes. But this is a quite old photo. Only this mosquitoes, you can see the white stripes clearly. Even in this mosquitoes, there are white stripes present, but here you can see clearly. See, these are the white stripes present on the legs. Here also you can see everywhere. So we worked on this. We did it continuously for a whole year. Means myself and Ubaid and all the other members also did it. So we have a result. So this was my result on the after the of 2017. I continuously regularly used to catch mosquitoes, then write it in the logbook, and then used to post it on the cube groups. And then we used to have discussions, so like that. According to this, from January to December, I had the data except the May month. In May month, I was not there in Mumbai. I went outside to my village. So I got the peak in March region of total number of mosquitoes. Whereas also there were many mosquitoes in September October region. Sorry, month September October month. And during this till April, I didn't got any Aedes. Like in the whole month, I used to get hardly one Aedes out of total. Total, I used to get about 400 to 500 mosquitoes per month. Daily, I used to get some around 20 to 30 mosquitoes, or sometimes less, sometimes more. But out of all these mosquitoes, only one or two used to be Aedes mosquitoes. But when the June, July, August month started, that time I found that the number of total mosquitoes reduced to a great extent, like from 500 to 400 to 500 mosquitoes. Directly came to around 14 to 15 or 20 to 30 mosquitoes per month. So like in June I got 20 to 30 mosquito per month. July I got 30-40 mosquito, like that. August I got some around 40 to 50 mosquitoes, like that. So there was a drastic change in total number of mosquitoes, but the Aedes ratio changed to a great extent also. That means out of 20 to 30 mosquitoes, around 80, if I got 20 total mosquitoes, then there used to be 17 to 18 mosquitoes which I found to be Aedes. So similarly the pattern continued till August, but September again the pattern changed a lot. September, the total number of mosquitoes started increasing, whereas the Aedes mosquitoes started reducing. So this was the data when I collected, and then we plotted the graph. So we got this graph somewhat near to my hypothesis as well as Ubey's hypothesis in the Aedes ratio. Obviously, this is the data from Cube Mumbai. We didn't had Cube Kanpur collaborators at that time. Now Hina ma'am would tell you about Kanpur things. So these are the mosquitoes caught by claptrap method in this 2021 when we resumed our work. So Abhijit caught 5 mosquitoes in Cube Navi Mumbai. This is a photo of that. Then I got 11 mosquitoes. This 11 mosquitoes also had one story that once I was just taking a bath during the morning time, 10 a.m. around. So that time in just 10 minutes, I got around 11 mosquitoes in just about 10 to 15 minutes. And the whole day when I tried to catch, I couldn't find that much mosquitoes. And again, there were none of them was Aedes mosquito. All were non-Aedes mosquitoes. As you can see, it is kept on the black background. Then Abhijit got one Aedes mosquito out of total 4 mosquitoes on one day. So that was the first time when we got Aedes mosquito in 2021 in Mumbai. Then after that, I got one Aedes mosquito some around 2 to 3 days ago. So that was our story in 2021. Now I would like Hina ma'am student to continue with.
Yeah, I would take up. Good evening, everyone. I'm Rahil Mudgal from Cube Kanpur. I've been working on mosquito model organisms since 2018, and here you can see the 3 different type of Aedes mosquito I have recently caught. And the first on the left side, you can see Aedes aegypti mosquito, which can be identified by the layer-like structure on its upper thorax region. Also, it is called yellow fever mosquito. It is the main vector of yellow fever, Zika virus, and dengue. And in the center, you can see Aedes albopictus mosquito, which can be identified by the straight line-like structure on its upper thorax region. It is also called the Asian tiger mosquito. It is the main vector of Zika virus, dengue, and chikungunya. And on the utmost right side, we can see Aedes vittatus mosquito, which was recently caught by me 3 days before in Kanpur. It was the first time that this mosquito was spotted here, and it can be identified by 4 or more dots on its upper thorax region. And it is the main vector of Zika and chikungunya virus. Now, identification of eggs.
On the mic. Yeah. So the eggs, here on the slide we can see 3 types of eggs, like the Aedes eggs. The Aedes eggs are basically, they are laying eggs which are sticking. They are glued to the surface, and the female lays egg on the inner surface of containers. And this was how we trapped the eggs on our OV trap that I mentioned in the introduction. So the first type of egg we see here on the slide is the Aedes eggs, and mostly the females, they lay egg in old car tires, empty bottles, vases, flower pots, and that's why Aedes is called a container breeder, because they need little water to lay eggs on. And the eggs basically look like black dirts. It's like whenever we are putting OV traps and we are locating black dot-like structure, like it's something like dirty is attached to it. And the eggs, they stick onto the surface where the female is laying egg on the containers where the level should be moist. Now, something interesting about Aedes egg is that they can survive for around 8 months in dry condition. That's why the moment they get water, they again start their life cycle, and that is the most dangerous part of the infection of the life cycle of Aedes. Now the second picture is of Anopheles eggs. Now Anopheles, female Anopheles, she lays around 50 to 200 eggs in one time. They are laid individually, and these eggs have been found to be floating on the surface of the water. Now, Anopheles mosquito is mainly a carrier, a vector of malaria. And the third type of eggs which we show here on our slide is the Culex egg. Now, these are like raft in a group. And raft means like it's looking like a small boat, and they float on the water surface. And the female Culex, she lays egg on fresh or stagnant water. It can be barrel, it can be puddles, ditches, small water accumulations. Now, it's very important for us to identify the different types of eggs. And we are putting emphasis on this because the concept of our home lab experiment this season was setup of OV traps. We had set OV traps, and when we had set OV traps, over to Samiksha. Samiksha Bhandari of class 10th, Allen House Public School. Over to Samiksha. Please take up and explain how did you do your OV trap setup? Because Samiksha.
Good evening, everyone. I am Samiksha from Cube Kanpur. An OV trap is a setup which is used to find out the current Aedes population. The OV trap was first described in 1966 and was initially designed for monitoring Aedes populations. Researchers found that if they provided artificial breeding sites, they could easily collect and study the eggs found in the container. Since the original OV trap was invented, lethal OV traps have been developed, which kill the larvae and the adult mosquitoes that enter. OV traps used for monitoring can detect Aedes mosquito populations, thus acting as an early warning signal to preempt diseases outbreaks. Analysis can be done on the OV trap breeding data collected weekly to identify the mosquito breeding hotspots and risk areas when there is a danger of high Aedes. Over to Vyom.
We lost you for some time, Samiksha. Can you please repeat what you were saying in the last this thing? The last two lines, please. I think we lost your connection.
Yeah, these analyses are used to plan vector surveillance and control operations. The extensive use of the OV trap in a community can be used in Aedes population control and effectively reduce the Aedes population. Over to Vyom, please.
Okay. So, how did we make this OV trap? We used waste bottles, containers, which were dark in color. And we have already discussed about the dirty looking Aedes egg appearance. Why we are emphasizing upon identification of three types of egg is different students. Pranat Kheria from Cube Kanpur had set up the OV trap, but he did not get his any result. But Samiksha and there were other students who had got Aedes eggs, which were, we got identified by our referral collaboration from other Cube groups, Dr. Sarita and Dr. T. Dilip helped us out. And here you can see this is Samiksha Bhandari's. She will explain her OV trap further, but I'm just briefing the initiation that what is an OV trap? It's basically a setup done to invite the mosquitoes, if prevalent in the area, to come and lay eggs. Over to the next slide, please. Who is it? Yes, it's Samiksha. Please take up. Samiksha, please take up. Are you there? Yes, please explain your experiment.
Yes, as a part of a home lab activity and mosquito surveillance study, three different jars with different waters, distilled water, tap water, and mud water were observed and were turned into an OV trap setup. The setup was made on the 9th of August 2021. It was placed near a shade where it got adequate sunlight. On the 11th of August, water was added in all three containers. On August 13th, the water was again added in the respective containers and no breeding was done. On the 15th of August, the position of the containers were changed and was kept near a plant in the garden. Water was added. On the 18th of August, soil was
Can someone else speak?
Yeah, I think she's explaining and we are losing connection, no issues. So this setup is by Samiksha Bhandari of class 10 and it was in the location of Lajpat Nagar. That's a very densely populated area in Kanpur. She had done setups from the 9th of August and she had taken three types of water. Like setup one had tap water, but she had used a common thing that is a dark container. Setup 2 had distilled water, and the third setup was soil containing. So again and again she is repeating on replenishing water. So this is an important thing, that if water is not available, then the breeding stops. So we have to check the water accumulation. That is what they have learned after setting up the OV trap. And finally, she got eggs, that is 20 Aedes egg on the 11th of August. And setup 2 was cooler water, where she did not get any eggs in that dirty water. So we had this conclusion from the study which the children did for a month and a half, that more Aedes eggs were being found in a dark container and in cleaner water. So the dirty water did not get breeding of mosquitoes, and we did not find too much of egg on the OV trap. And over to the next slide, please. Yes, Vyom Gupta, please take up.
Good evening, everyone. I'm Vyom Gupta from Cube Kanpur. I had set OV traps with two different water samples as a part of our home lab activity. The objective of setting these OV traps was to determine the type of mosquitoes found in my locality, due to the fact that they are the vector of many diseases, and also to bring awareness amongst the common people to check for breeding sites. The OV trap I built was completely eco-friendly and a frugal innovation of mine, as it was made from materials that were no longer needed by anyone in my family and were readily available. Hello, am I audible now?
Yes, yes, you're audible.
It helped in providing simple experimental methods. So for the materials required were a ruler, a cloth, a rubber band, and a plastic container. All of them were readily available to me. The procedure of setting up the OV trap was straightforward and uncomplicated. Anyone could do that. I took a ruler, I wrapped it up with a cloth, and then tied them together with a rubber band. I then filled my first plastic container with water that I obtained from my water cooler on 9th August, and the second one with tap water on the same date. I kept both my OV traps in an open area near the stairs. I had to fill my I had to refill my containers from time to time as the water evaporated. This was a crucial part. After a certain period of time, I observed and found some black structures formed. I immediately reported this to my mentor, Hinamam, and after some consultation from a cubist belonging to Kochi, Kerala, they were identified as Aedes. Now, for my experience, prior to doing this experiment, I had no idea about what a OV trap was and why mosquitoes would lay eggs in the apparatus I put together. And like a toddler who discovers something new, I was over the moon when I found these dark structures formed. In my first OV trap, which contained water from my water cooler, I found 29 eggs and 17 eggs in the second one, which had tap water. This confirms that Aedes is breeding in my locality. And for the note, carrying out this experiment at home with help from my mentor Hinamam was an exhilarating experience for me, and I also learned numerous new things as well. Now over to Abhijit Bhaiya for the concept map.
Okay, so the concept map will speak upon. So,
So
mosquito is
yes, so
connected.
we continuously try to connect our organisms with what we study in our home lab or what is the organism that we are using. So with mosquitoes, we can connect it with a curriculum, like we can understand life cycle with the help of that, and in life cycle we can study the eggs, the larvae, and pupae, and how it get converted into adults. So yeah, this we can understand. Then we have pollination. In pollination, we come across cross pollination and dispersal pollination. So mosquitoes are also the pollinators for various plants and uh, uh, flowering for that matter. And then we can study about diseases. Our main focus is like we are focusing on diseases, like how to reduce the number of cases that we are coming across. So that is one of the main things. So we can study about diseases like dengue and yellow fever, malaria, and chikungunya. So this all we can study with the help of that. Then we have frequency. When we trap the mosquitoes with the help of clap trap method, then we maintain our log that each day how many mosquitoes we got. So we mention in the log as like date, then number of Aedes mosquitoes, then non-Aedes, and then what was our difference in that. So that we the statistics as well. Then we can understand immunology. And when we get diseased with a particular kind of disease, then how our immune system responds to it, understand. Then through classification and taxonomical studies, we can understand about the how a classify how we can classify the biology with the help of that, like how insects are studied in that category. Then we have Nobel prizes, like obviously we search for the previous work that has been done on our
Abhijit, excuse
Yes, Hinama, you're trying to speak.
yeah, actually, mosquito has 4 Nobel Prize bagged on its as a model organism. This is very interesting.
Yes, so we have Nobel Prize. We can give you the literature on that. So Ronald Ross in 2002, he won Nobel Prize in medicine and physiology and medicine on the discovery of transmission of malaria. So then we have in 1907 we have Alfonso Laveran. He was awarded the Nobel Prize in physiology and medicine in discovery of parasitic protozoan as a quadratic agent for infectious diseases like malaria and all. So it was awarded to Alfonso Laveran. Then we have Paul Mueller. He was awarded the Nobel Prize in 1948 for the insecticidal qualities and the use of DDT in the control of vector diseases such as malaria and yellow fever. So DDT can be used as a control agent to control such type of disease. Then recently in 2015, Yu was awarded the Nobel Prize in Physiology and Medicine. I think that part she was awarded the Nobel Prize in discovering the anti-malarial drug that is artemisinin. So we can review this all literatures with the help of that. Then we can study about microevolution. We come across these terms in our curriculum in a degree college and like that. So we study about adaptation, then genetic variation we can study. Then we have inheritance pattern, then also evolutionary biology. Then we can study about habitat, what kind of habitat mosquitoes lay their eggs and what kind of things they prefer. Then, as a part of ecology, we have ecology in our curriculum. Then we can study about environmental interaction, how the behavior of mosquitoes changes or the egg-laying pattern changes with respect to increase in temperature, decrease in temperature, then salinity, then water, and also that stuff. Then we have tropical secondary, tropic level secondary, then that we can study. Then food chain. This mosquito larvae, they eat protozoans and bacteria, and that when they're in larval stage, they eat and they feed on them. So with the help of that, we can connect it with the food chain and animal association. Also, rotifers and nematodes, they also sort of feed on the mosquito legs, and that we can study with the help of that. Then, of course, we have control measures. Like this is one of the major aspects, like how we can control the mosquito spread. Then we have pest control. We study about the biological controls. We can use fishes. Various kind of fish eat mosquito eggs and larvae. Then rotifers can be connected with that. And also cyanothrips can be different bacteria. Bacillus thuringiensis, this particular kind of bacteria can be studied with that. So also this bacteria eats up. They can control the level of mosquito spread. Then we have chemical sprays. The civic bodies in our area, they spray. They do fogging in order to control the mosquito spread. This is the concept map or the context which we are understanding mosquito or we are studying mosquito. How we can connect it with our curriculum. And that is very important. And this is our sorry. Then we have mosquito collaborators map. It is very interesting to have collaborators, and it also helps us to get collaboration from different parts of the country. And we have Dilip sir from Kerala, then Shankar sir from Kerala, then we have Ashwati, then Anand sir, Kiran Saida from Mumbai, Gaurangi, then we have Hina ma'am from Kanpur, and Vyom, Samiksha and their collaborators. Then we have Raj from Delhi, and then we have Batul ma'am and Ban, and also we have Vishika from Assam. So this is a kind of collaboration that we are discussing our mosquito projects and understanding through discussion in Chatshala. We are understanding it better and continuously try to develop it. And I hope that this collaboration map can be extended to more parts. Nowadays we are also getting more collaborators. Like today in the Cube WhatsApp group, Seema from Cube Mumbai posted the photo of an organism, and that pretty much turns out to be the larvae of the mosquitoes. And we told her to discard the possible breeding sites so that mosquito won't be breeding in that part. So we are expecting more collaborators, and through this we are spreading awareness, because that is one of the main objectives. Because it is not just the duty of our civic bodies to control this, but it starts with personal level. Like when I'm studying this, I will educate and I will tell my family, then my neighbors and my relatives. So it starts from very individual levels. That is a kind of awareness that we are trying to develop among all of us. So now we are done with the presentation and we are open to the questions now.
So, in one of the slides, you have actually shown the life cycle of the mosquito, and it was the life cycle of Culex. So, there's a question by Sarita ma'am that what is the difference between the life cycle of Culex and Aedes? This is actually the life cycle of Culex. So, what is the difference between Aedes life
Can
cycle?
I take it up, Seda?
Yeah. Yes.
Actually, this is the life cycle of Culex because you can make it out by the egg raft. I just told in the presentation that the Culex lays egg in the form of a raft, and that is how we identified that the mosquito breeding. That is what is the concept of ovi traps. We are not culturing mosquitoes. We are just trapping the eggs to identify which type of mosquito is breeding. So in this life cycle, this is a life cycle of Culex. It has egg rafts because Culex lays eggs in rafts. They are glued together and they sail like a small boat. But in case of Aedes, they will be glued to the surface. The Aedes aegypti has 4 stages: egg, larvae, pupae, and adult, and the entire life cycle from egg to adult. It takes approximately 8 to 10 days, and the pupae develops into an adult from around 2 to 3 days. So basically, what happens is if you just identify the eggs, in the case of ADs, they will be glued to the wall surface or the container, because normally ADs are found in small water containers and all. So that is one of the difference, and the other difference is that the larvae of ADs and the pupae, they keep coming to the water surface to breathe. So that is also how we identify. We have a lot of pictures of those samples, but basically by the eggs, and I hope I have made my explanation clear. And Aedes aegypti takes 7 to 10 days. It takes 7 to 10 days to develop into an adult from
As
an
we
egg.
as we are discussing about the egg, can we go to the slide that actually
Yeah, sure.
describes
If you,
the eggs?
yes, who's controlling the slide?
Yes,
Abhijit?
ma'am.
Orangi, can we go to the slide of the eggs, please? If you look at the eggs, the Aedes eggs, just before this, yeah, this one, yeah, this, the
There is
Aedes
a
eggs.
question with respect to the eggs, that the Aedes eggs and the Anopheles eggs looks very similar, and Aedes eggs is just some are scattered and some are not scattered. So how do you differentiate between Aedes and Anopheles eggs?
Because in case of Anopheles, it's never in groups. It's mostly they are floating. Individually, they float on the water surface. But in case of Aedes, we find the eggs attached to the surface. Like if you see, they are mostly attached to the surface. Like if there is a wall, side wall, they are attached to the side wall. Like whatever studies we have done, we have always found Aedes egg on the wall surface where the water is close to it. Neither it is dry, neither it is too wet. So that is the main difference. But in case of Anopheles, you can see that they are not in group. They are all single, individual, and they'll be floating on the water surface. Okay, and sometimes it happens that that these Aedes aegypti eggs, they do, they are always present near the water containers and the water line, and they can survive this. Even if it is dry, they survive for a long period of time. That is a very important thing. That's why the cubists who were doing the ovitrap setup, they were needing to replenish the ovitrap setup. Small amount of waters, the female comes and lays eggs. Bowls, cups, fountains, tires. You can show the ovitrap picture, please.
Okay, so it is a very good presentation. So I have one question regarding, so in the presentation we saw that what are the possible breeding sites for mosquitoes to lay eggs. But what are precautions, what all precautions we are taking? Because we are also involved in the OV traps and we are seeing some breeding sites. How do we stop that?
So the following precautions we should take, that we should not go in a particular region with a lot of mosquitoes just for our project. Yeah, for if we are trying to keep the OV traps, we can in our house itself, where we are finding more mosquitoes, we can keep the OV traps, or in the nearby areas. But take care that the more mosquitoes are not biting you, and apply odomos or any repellent, mosquito repellent on your body. And if you are seeing other than OV traps, if you are seeing normal water which is collected in any thing like bowl or any planting, that what we call that matkas. So take care that if you see, then just take an initiative to throw it away. If you see wherever in your nearby area, in your house, or in, in your surrounding, then just throw that water, collected water. um, and even in ovi traps, after you noticed the eggs, after you count the eggs, you take the photographs, just clean it off with Dettol or some infectant, whatever if we have home. But Dettol is mostly available at everyone's place. Just put some Dettol on it, and then clean that cloth thoroughly, and take care that the eggs are completely dying before washing it. So apply any disinfectant or Dettol or things like that. So these precautions you should take, and you should also create awareness in all your neighbors and family members that they should stay away from mosquitoes. Even if you are doing mosquito project, then it is not that you go in mosquitoes and trap the mosquitoes, not like that. Just the mosquitoes which are coming to bite you, that you're killing it, that just we need, that just collect the photographs like that. So these are the precautions we should take.
So one important question with regards to awareness is the water the key to actually get rid of the mosquito? Like if you don't keep water um, in any container, don't allow the water stagnant water to
Yes, stagnant water is an very important key to get rid of the mosquitoes. We should take care that there is no stagnant water in your houses or nearby areas. I know you can't go and clean the puddles, but wherever you see water stored in your house or nearby areas, just take the initiative to throw it and not allow the water to remain stagnant.
Another question with respect to mosquito is okay, we can use repellent and stuff like that in order to um, uh, kill in order to basically save ourselves from mosquito. But you in your 2017 study that you were talking about, it talked about the equinox as well, that your hypothesis is that particular time we'll get more number of mosquito, and September equinox was not very far. And there's a YouTube comment on that what is your observation of number of mosquitoes in September equinox, and equinox as it was very recent
In 2017 or in 2020 when you're talking,
in
Sarah?
2020. I guess the person meant 2020 because the comment says that it was very recent, so I assume it is he or she is talking about the recent.
In 2021, we just resumed with our work. So in September, we tried catching the mosquitoes, and the total number of mosquitoes were less in my area at least. And I only found 2 to 3 Aedes mosquitoes all over this month. Even Abhijit has just found one Aedes in September month. And I would ask Hina ma'am for her data.
Um, we had done our survey in 2018 and 19, and basically we had shared our data in the last cube meet that we had observed more mosquito breeding in September and October, and we had got Aedes mosquitoes. We had collected pictures of around 33 mosquitoes per week, and we got more mosquito population in between September to October. So that was, I think, the fall. That is the fall time, and we also got a good population of mosquitoes in June. Okay, so these two seasons we had a good study time, and that is during the summer vacations when we had conducted a survey of sample collection. That is in the year 2018 and 19. So I had one thing that basically when the temperature is either too hot or it is too cold, then the breeding is decreasing. But if the temperature is moderate and the humidity is there and temperature is moderate and then availability of water is also there, then we have observed good breeding, and we have got a lot of samples. That's it.
So that made so much sense that okay, why Gaurangi and other collaborators were guessing that okay, rainy season would be the time when there would be more mosquitoes. So I think with that, great work mosquito group and other groups as well. Great presentation, and I think looking forward for so stay tuned. National Meet has not ended yet. Tomorrow we'll be having presentations on Earth. On cardamom, butterfly, snail, zebrafish, economics, with different questions, different context to curriculum, different. So it would be we are it would be definitely very very interesting. So looking forward for you to join the third day of our national meet. Take care. Thank you. I think with that, yeah, good night, everyone.
Yeah, thank you so much. And we are looking forward to see everyone tomorrow as well, which is your final day of national meet. Thanks a lot and good night.
Thank you.